morning, everybody. Hello. Thank you so much to be here again for the next day of our workshop. We have a terrific lineup of speakers. As yesterday, I hope you enjoy the whole afternoon with us. And let's learn a lot about bioinformatics and applications today as well. So I pass the word to Diogo Figueiredo that will introduce again, for the record, the Anchor program. Thank you, everybody. Diogo.
Yes, thank you, Professor. So welcome, everyone. Like Professor said, we are here today live on YouTube for the second day of the Biotec, Bioinformatic Tools and Applications Workshop. We'd like to thank everyone who participated yesterday. We had great presentations, very insightful, and I'm sure today's presentation will be as well. Before we get started with the presentations, I'm just going to make a small introduction. So my name is Diogo Figueiredo. I'm a consultant at SPI, and together with Professor Krieger and then Professor Fabiano, we have been developing this today workshop. Just to give you a little bit of context, this workshop is one of many activities from the All-Atlantic Cooperation for Ocean Research and Innovation Project, which is called ANCHOR for short. This project is a project funded by Horizon 2020 under the umbrella of the All-Atlantic Ocean Research Alliance, and its main goal is the implementation of the Belem Statement with the aim to upscale the research and innovation cooperation within the Atlantic Basin, also to better understand and sustainably manage the Atlantic Ocean as a whole, and to see the basis for a healthy future large-scale joint Atlantic marine activities. So today's workshop is part of the joint pilot action AA Biotec, which is short for the All-Atlantic Marine Biotechnology Initiative. This joint pilot action aims to foster collaborations across governments, academic, private sectors, mainly in the field of marine biotechnology. It also aims to create mechanisms of technology transfer and promote the development of new marine biotechnology companies at the Atlantic Ocean level. On behalf of the Anchor team and everyone involved in the organization of the joint pilot action and this workshop, we will just like to let you know that in today's event we aim to be inclusive, embracing and engaging, and also promote the exchange of knowledge across the entire Atlantic. I'll be here today along with Juliana and the other staff as well to help Professor Krieger with the workshop. Any questions you have or anything you need, just let us know. And if we can be of help, we'll be sure to let you know and take care of it, and also pass your questions to the presenters. I'll also take this opportunity to inform you that the next event of the Biotech Mar Joint Pilot Action will take place on June 27th and 28th, with the first day of this event focusing on aquaculture, and the second day, June 28th, will function as a sort of a startup accelerator. So just to conclude, I'd like to thank everyone for your participation, and I wish you all an excellent event. I'm going to now pass the word to Juliana. Juliana, go ahead. Once again, thank you.
Thank you very much, Diogo. Okay, so welcome everybody to our second day of event. I hope you're all ready for a very exciting schedule for today. Our first speaker is Diogo Tschoeke. Dr. Tschoeke has a bachelor in biological science from the Federal University of Santa Catarina, a master's and PhD in computational biology from Fundação Oswaldo Cruz here in Brazil. He has a lot of experience in genetics, acting on the following subjects: bioinformatics, protozoology, evolution, comparative genomics, and metagenomics. Currently, he's a professor at the Biomedical Engineering Program, COPE Institute at the Federal University of Rio de Janeiro. He will be talking about the mining genomes from metagenomic data, introduction and application. Thank you very much, Dr. Diogo Tschoeke. It's a pleasure to have you here this morning.
Good morning to everyone. I would like to thank you for the invitation and say that yesterday I watched all the interviews and the seminars, and say that was an amazing talking yesterday also, and hope that today all the people who watching us could enjoy our presentation. It's everything okay? Okay, you can see me. The presentation is here, so let's start. One moment. Okay, so my idea here is talk a little bit about genomics and metagenomics, assembled genomes, which we call MAGs. Talk a little bit about the methodology and then an introduction to this field, and show to the audience applications of this kind of initiative and analysis. So my agenda today, I will share a few slides about an introduction to metagenomics, what is a metagenomic field, and the recent advances in the field. And the second part of the presentation, I will talk a little bit about the methodology to access these MAGs and how we analyze a metagenomic data, and how we can measure and how we can know if a MAG, if a metagenomic assembled genome recovered from genome, it had a good quality or no. And in the third part of my presentations, I will deliver some examples and applications to concerning this kind of analysis. So metagenomic data, and generally speaking, when I talk with my students and the people who work with me, I generally ask 3 questions. What is metagenomics? What is it for? So what is the function and how it works? And as we know, metagenomic data, we access generally the microorganisms. And the microorganisms, who are these microorganisms? Generally, we access bacteria, eubacteria and archaeobacteria, generally viruses we can also access using this metagenomic approach, but not only limited to these 2 domains of the life, but we can also access fungi and protozoans and other eukaryota. Also, if I don't get me wrong, Matt yesterday talked a little bit about how, for example, we can access fungi and other eukaryotes using metagenomic approach, which was a very interesting talking also. So the microorganisms are everywhere around us. They are spread even in our body. That also an interesting book that the title is 10% human, or 1% human. And what we can say is that, for example, we have more bacterial cells in our body that our proper cells, myself body. So, for example, if we will study, for example, a milliliter of seawater, we can find more than 160 species in 1 typical milliliter seawater. Typical seawater is the seawater that the temperature not so cold, not so cold, not so hot, and not too much salinity or not too many salinity in the field. And the soil, for example, and depending on the diversity of the soil that we are studying, for example, we can find around 60,000 up to 40,000 different species in a soil of grass, or depending of the environment that we are studying. And this is an intriguing question, and this is the concern of the metagenomic studies. What are these microorganisms doing? And of course, the question is, who are the organisms that are there? But what are these microorganisms doing? It's vary a lot depending of the environment. But the environment, it's a greater reservoir of genetic diversity. And when the metagenomic analysis has grown years ago, 20 years ago, we noticed that less than 10% of the microorganisms in the planet was already characterized and described. So the metagenomic analysis, or the metagenomic, I consider a field of the genomic, of the big genomic science. The metagenomic data, or the metagenomic analysis, try to access all the diversity that we can found in a sample. For example, must be water, must be can be a soil compartment, or other environment also. So the term metagenomic comes from the Greek. It's beyond, and so the idea of the metagenomic analysis is study more than one genome at the same time. The idea of meta is the same that we use for a meta-analysis. And so I do this parallel with my students. So when we do metagenomic analysis, the idea is the same of when we do a meta-analysis. Then the idea is study more than one variable if we consider a single genome a variable. So the idea is to study a collective of genomes in an environmental sample. And when I say environmental sample, it's a very broad idea of environmental sample. For example, if we study, for example, there are many studies, for example, that try to access the diversity of a gut of a plenty of organisms. Also, for example, we have some studies that we access the diversity of the termites gut, for example. And so the idea of the environmental sample, it's a very broad idea, not specific environment itself. And the term was introduced by John Haldeman in 1998. And sorry. And the idea, compared with the genomic, as the genomic, we study a single lineage, unisolated, for example, the classical approximation of microbiology, and the metagenomic data. The idea is to study a community of microorganisms. And when the metagenomics start, generally we start using 16S analysis, or targeting, and analysis targeting some gene markers, for example. 16S is one of very famous and useful for bacteria analysis, for example. But as the field was evolving, and the computational analysis also was evolved, we can access now not only the microorganisms itself, only small pieces of its genomes, but now we can also recover full-length genomes from this metagenomic data, and I will show to us how interesting the results of this metagenomic assembled genome analysis could show to us. I like very much this slide. Sorry, they are in Portuguese. I forget to translate. The idea of genomic study is when we use a pure culture. For example, here we have a petri dish, and we have theoretically only one organism. Of course, there are some cultures that are not axenic. We have more than one different species, but the idea is that we have a limited diversity in our petri dish. However, when we study a microbial community through metagenomic analysis, the idea is access theoretically all the diversity that we can find in this specific field. And there are a work for Luxley and Lennon in 2016 that they estimated that for each microorganisms that we know, there are more 100,000 species to be identified. So with the metagenomic analysis, now we are starting to know this hidden biodiversity. So there are some synonyms for the metagenomic analysis. For example, there are some people that call it an environmental genomics. The idea are pretty the same. When the researchers know years ago that are impossible to cultivate all the microorganisms, the metagenomic received an impulse, and now there are a lot of works in not only in Brazil but in the world, accessing the biodiversity using metagenomic data. So the idea is study nucleic acids, genomic DNA from unknown organisms. So the idea of, and so why metagenomic analysis is so important? Well, it's a very good methodology to study microorganisms. First, because the analysis does not involve cultivation. So the cultivation of a microorganisms generally, it's a very selective pressure, and many times we select not the most abundant organism, for example, that we found in an environmental, but we select the microorganisms that have the ability to grow better than the other microorganisms in our culture. So this does not involve cultivation. Theoretically, I say theoretically because, of course, we generally cannot access 100% of the diversity in a sample. But the idea is directly extract the DNA from the environmental. It could be water, soil, or air, for example, and allow us to map the biodiversity of a sample of a location and sequence all these DNA data. We can also, for example, access genes involved in antibiotic resistance, for example, or genes that could be interesting to biotechnology industry. And there are many examples of products that were generated by this metagenomic analysis. So there are here a short example, for example, marine environment. The idea of metagenomic is directly extract the DNA from this environment. So we sequence this DNA from this environment, and in principle, we can access 100% of this genetic diversity. And when we use the traditional approach, and I don't like this term very much, but when we use the traditional approach, we cultivate the microorganisms that are present in this sample. And after that, we isolated the DNA and sequence this DNA in an approximation that could be the genomic analysis, for example, proteomic, or there are plenty of analyses that we can do with this data. So depending of the environment, for example, there are environments that we can cultivate, for example, 10-20% of the no diversity. And what is interesting is nowadays that the metagenomic analysis are very developed and show us very interesting results. There are many studies comparing in the same sample what we can recover using the traditional approximation, using cultivation, and using the metagenomic approaches. And for some studies, for example, the cultivation could access 1% of the biodiversity, and the others studies show us that, for example, we can access almost 20% of this diversity. And so, yeah, we have a long way to do perform until we get this interesting analysis. So I like very much this picture, and there are interesting questions, and there are these two main questions, I guess, that metagenomics try to answer to us: is how is there, and what are you doing? I like this picture because if you look distant from the screen, generally people see a O. But when we take a closer look, if you get closer to the screen and take a look in the details, you will see that this picture is formed by many small circles. And these small circles, when combined, passing the idea that this picture is a O. And the idea with metagenomics is the same. We try to identify what are the organisms that are in this location and what these organisms are doing there. And I guess this analogy is very interesting because the idea of metagenomics is that we want to know how this sample, how this environment works, is the final idea. So when we study some parts of this sample, of this environment, we try to assemble it in a big picture, as we can see here. But I will, as we are in a bioinformatic workshop, I will talk more concerning the bioinformatic analysis, but of course there are many wet lab work to do with this metagenomic data, and depending of the sample and the nature of the sample, we have different ways to process the samples. But how is it done, this analysis? So the idea is that we have a community, as I showed previously, and we extract the DNA. But when we extract the DNA, we don't know exactly which piece of DNA belongs to which species. This is the idea. So we are flying blind without seeing what are these hidden diversity that we look in this data. And through bioinformatic analysis, we can identify, if we are working with a double GS analysis, without working with a specific gene, with using a whole genome sequence of this metagenomic data. The idea is try to identify through bioinformatic analysis, generally, for example, using tools that use similarity, for example, like BLAST, BLAT, Diamond, or this analysis. And we try to identify every piece of DNA. We try to identify to whom these pieces belong and what are the function of this piece of DNA. So with this analysis, we can try to identify and create a model of how a determined environment works. So this is the idea. So for example, which organisms are present in this environment? There are a very interesting work, for example, that we published years ago of try to identify and try to look in an explanation for why the algal mats in the Abrolhos Bank are so successful. And we are looking, and when we look to this data, the microorganisms present in these algal mats, and algal mats, no, sorry, it is, the algal mats, we found that the microorganisms they're working as almost a closed system. This is very interesting work also. And for example, you can try to answer how the phosphorus cycle works in this environment, or for example, how an sample and an environment respond to pollutants, for example, in the environment, or how specific cellulase, for example, are degraded. So when we looking and analyzing this metagenomic data, we must to consider, for example, that a specific taxonomic rank, for example, a phylum or a class are in the highest taxonomic level are very rare, very, very, very rare, for example. But when we go deep in the taxonomy level, for example, in the lowest level, like genus and species, are often common to find specific genus to a specific environment or a specific sample. It's more common than to what happened with these high taxonomy levels. And also, for example, if we have a very specific ambient, like for example hot springs or an environment with high salinity, generally we have less diversity than ambient and environment that are less restrictive, for example, like a soil or fresh water or salinity or a sea sample, for example. And I will focus on the results and the idea of the metagenomic data and how we can recover the mags from this environment using this pipeline here, that we do the sampling, extract the DNA, sequence this DNA, generally using Illumina sequence, but there are very interesting works now with a long read sequencing machines, for example, like PacBio and Menio, show us interesting results, and we access theoretically all the diversity that we can find in this environment through sequence. Comparing to the first approach of metagenomic data, when we sequence a specific gene in this environment, for example, like 16S or 18S. And the metagenomic analysis is related to many advances in our field. And we have, for example, many improvements in bioinformatic tools to analyze this data, because 10 years ago, not 10 years ago, but 15 years ago, for example, a sequence machine delivers, for example, 1 giga of information per each time that they are running. And nowadays, for example, the highest throughput that I know is the Nova 6000 that delivers us almost 6 terabytes of information. And the metagenomic analysis demand an improvement in the computational field also, not only how we analyze, but improvement in the algorithms, in the structure to store and analyze this data. Because most of the bioinformatic, most of the metagenomic analysis is based on, it's computer-based, made by bioinformatic tools. There are many of these tools that are used to genomic data, to analyze genomic data, and to analyze metagenomic data. And of course, there are some problems. For example, as metagenomic data nowadays, most of the analysis of this DNA sequence are accessed using small read sequences like Illumina. Assemble these small reads are not so trivial or not so easy to do that. But there are many programs that nowadays work better for metagenomic data than, for example, 10 years ago. And this is an overview of a metagenomic workflow, not specifically related to the MAGs. I will talk about how we access these MAGs, but a typical overview of the metagenomic workflow start with the sampling. We need to select an environment to sampling, extract the DNA and sequence the DNA. This is more related to wet lab, using, for example, kits of the DNA extraction, library construction, and otherwise. And once the DNA is sequenced, we start to work with the computer using bioinformatic tools to access this diversity. And once we analyze the quality of the sequence, we can, for example, assemble this data, or we can directly try to do a gene prediction of this DNA data. And once we know what are these genes, or where start a gene and where end these genes, we can go to the annotation of this data. And for example, the assembled gene prediction and annotation is, there are many studies that call this part of the analysis that we call it the binning. What is the binning? Binning is the idea to identify the sequences, to whom they belong, and what the sequence do. And after that, we can do some statistical analysis and store this data. Of course, this is a very general over workflow. For example, there are some particularities if you must work with microbiome, human microbiome, for example. We need to subtract the sequences from the host, on our DNA, in this case, for example. But the idea of analyze a workflow, a metagenomic data, is run through this workflow. And what is the idea of the metagenomic assembly? There are plenty of programs. Here I show 5 programs, but I will show in the next slides. There are more than 10 programs to perform metagenomic assembly data. And the idea is here we have a metagenome that it's formed by many of small pieces, like a puzzle. And when we assemble this data, the idea is we try to recover the full picture or the full genome of the bacteria or archaeobacteria or viruses that are present in this metagenomic data. But there are some issues that we need to address. For example, the diversity of the sample. For example, if we are working with a very diversity sample, for example, this is a rarefaction curve analysis. This is the case of the first example that I talk. When we working with a very diversity sample, we need to sequence many data, a lot of sequence to know the full diversity of this environmental, and also to be successful to recovery these mags from this analysis. Why? Because when, in this case, we are sequencing only a small part of the sample, we stick in this case, for example, that we have, or that we sequence only small pieces of the genomes. And this second idea here, the second curve here, this blue curve, it's a sample, for example, that we are closer to the real number of the species. And so in the second case, it's more likely to recover mags and complete mags. And in this third case here, the green one, the green curve, we increase the number of sequences, as we can see here. We increase the number of the sequence of the sample, but the number of the species does not increase. So in this case, we say that this sample reached the plateau of the diversity. And in this specific case, for example, we are more likely to get more mags and more complete mags from this sample than, for example, this first curve here. And so we need to consider and take in account the biodiversity of a sample also, because, for example, if you have a small coverage to a high sample diversity, probably we will have problem to recover full-length genomes from this metagenomic analysis. For example, this is a work that Luciana was our PhD student performed. She worked with water from the Campus Basin region, and we are trying to study and looking for the oil biodegrading repertoire of the samples. And as we can see here in these rarefaction curves, most of the samples are near closer to reach the real plateau of biodiversity. But in this case, for example, we obtained only 12 mags from these samples that reach our criteria of quality of a mag. But how we can recover, for example, how here in UFRJ, in our group, how we can access and recover metagenomic assembled genomes from the metagenomic raw data. So generally, we use a program called PrinSec to analyze the quality of the sequence and remove the sequence that don't achieve our quality criterion of quality. We assemble the data, for example, with megahits or metaspades, and once we assemble this data, we map the raw hits using, for example, a bow tie, which is a alignment program. We map the raw reads in the scaffolds that we generated with megahit or metaspace, for example. And using the information of the depth of each contig in our samples, together with the scaffold generated with metaspace, and submitted these two information to MetaBet. And MetaBet will, it's almost like will try to identify profiles and gives you putative metagenomes recovered from this genomes recovered from this metagenome data. And after that, we need to check the quality of these metagenomes and looking for parameters like completeness, contamination, and phylogenetic position. And when a genomic when a genomic satisfy our criteria of completeness, contamination, for example, we can annotate these genomes using, for example, RAS or a PROC or another annotation to specifically identify the genes and the taxonomic position. So this is a metagenomic assembly program. This is a very interesting review of the of tools for generating mags. There are, for example, programs here, Metaspace, Megahit. There are here in UFRJ the program that you use more, but there are plenty of others. And this there are very interesting work. I guess it was one of the that can able to recover nearly 8,000 metagenomes. And this work shows us the potential of the metagenomic analysis concerning the recovery or retrieve the mags from this analysis that, for example, propose many different phylum here in our tree of life, for example. So it's a very interesting work. For example, here is in red are representatives of for example, a specific phylum that we do not know what are our closer sister concerning if it cultivable or not. And when they looking for specifically archaea genomes, this work was be able to identify, for example, here new phylum here and here, two new phylums, and one is that is closer to the Lochiarchaeota, which is considering a phylum that is sister to the eukaryotes. Here, I will talk a little bit about this. But, of course, there are two main ways to recover the mags. This is the first way to recovery, but here we use a reference-based genome, and we cannot discover new genomes because we are mapping the biodiversity in those genomes, and so we will stuck in the de novo discovery. For example, we cannot be able to find completely new genomes, but when we use the MAG itself approach, which is explained in the next few slides, we can able to recover and discover completely new genomes in this environment. And here there are many programs to do these metagenomic tools. Each of these had a different way to identify and classify the metagenomic assembled genomes. But when we started to work, for example, I started to work with MetaBatch, which is MetaBatch and Concotter are the two most famous binning tools for recovery metagenomic data. So this is the MetaBat and MetaBat 2. The idea behind this program is that we assemble this data here. We have samples from metagenomic data. We assemble this data, and when we finish the assemble steps, we submitted these scaffolds or these contigs to the MetaBat, and the idea is calculate the tetranucleotide frequency of each contigs, and also calculate the abundance of each contig in the different libraries that we are working. And the program used these informations of the abundance per library for each contigs and the tetranucleotide frequency for each contigs, and iteratively forming these bins. And so there is a statistic model, probabilistic model that modelates this distribution, and using this information, the MetaBat 2 or MetaBat gives you in the end of the analysis the possible bins or the possible genomes recovered from these metagenomes. But why also the abundance is important to this program? Because contigs with similar coverage are likely to belong to the same genomes. This is the idea that we also use as a parameter the abundance of each contig. And the tetranucleotide frequency is very important because, for example, when you construct an Euclidean distance using the tetranucleotide frequency of each contigs, we found that, for example, when we have a distance of more than 0.5, this Euclidean distance, the probability of two different contigs belong to different species is almost 100% of probability. And of course, the size of the contig is very important also. For example, it's not so common in metagenomic assembly data. We have a contig so big that is, for example, this is 5,100. But as the idea is, the bigger the size, the bigger the size of the contigs, higher is the probability of the program to classify correctly two different contigs belonging to two different species. And why MetaBat? Well, MetaBat has a very good results, as we can see here. Is the paper from 2019 comparing the MetaBat with other binning programs. But when we get the results of these contigs, of these mags, is the end of the analysis? No, generally not. We need to access the completeness of this genome and the contamination, for example. And so this work of Parks from 2014, they are propose an uncontrolled vocabulary to classify genomes. And so, for example, if you have a genome with more than 90% of completeness, these genomes are near complete, and if the genome has possess less than 5% of contamination, they are classified as low contamination level. And there are very interesting terms that we already use in our analysis. So the idea of CheckM is try to identify the lineage of this specific genome, for example, to which, for example, which phylum or which family belongs to this contigs, to this genome, and after that they looking for genes that we must know that must to be present in this genome. And considering this information, the CheckM give us an information of completeness and contamination. And of course, higher is the completeness and less is the contamination, more accurately will be the parameters that CheckM deliver to us. So this is what this figure show to us. But okay, I talked about metagenomics, what is metagenomics, how to access these mags, how to evaluate the quality of these mags, but what specifically this analysis of mags in a metagenomic analysis can show to us? This is a very interesting manuscript that is specifically looking for archaeobacteria in a region near between here the Greenland and the Norway. It's called the Loki Castle. This is why the phylum is called Loki Archaeota. And using, of course, very different metagenomic approaches, as we can see here, this group was able to identify a genome that is an archaea genome. As we can see here, it's an archaea genome, but have components that are specifically, for example, to the eukarya genomes. Like, for example, some ribosomal protein, GTPases, and here we look in the identification of this phylum Loki Archaeota and the genomes and genes that are identified in this specifically genome, and almost all specific genes that differentiate eukaryotes from prokaryotes was identified in this genome. So this is I consider a very interesting and very, very good work to explain how we can use a combination of genomic and metagenomic analysis. And this is other work from Hug and collaborators, and was published in Nature. This red circles here is the bacteria that was identified using metagenomic analysis, and here is the Loki Archaeota field, Loki Archaeota leaves. Here it's closer to the eukaryotes. And now, I almost running without time, and now I will talk about a little bit our results that we can able to identify some mags in our metagenomic data. And this is the work that I showed to us the previous slides concerning the rarefaction curve analysis. And here, Luciana performed an analysis through almost 60 days, and she get water from the surface of the campus basin should get water from the maximum chlorophyll region and from the bottom of this location. And using metagenomic analysis, we were interested to know what is the differential abundance of, for example, these 5 specific genomes, genes that are related to oil degradation. And as we can see, for example, there are some bacteria that increase in abundance between the days when we looked for samples that are incubated with water and oil here, for example. And when we're looking for the genes, specifically the genes that degrade these oil components, we observe, for example, an increase here in the abundance of some of these genes related to, for example, degradation of alkanes, anthracenes, naphthalanes, and other these genes that are related to this specific analysis. And when we assemble these genomes and try to recover these mags and identify what is these mags, for example, we identified novel metagenomic assembled genomes for example, Alcanivorax, from Oceanicalius and Thalassospira. And it's interesting to note that almost all these genomes, these recovery genomes, possess genes related to oil degradations. It's a very interesting result that we get from this analysis. And as an extension of these previous works, we analyzed the metagenomic data from a campus basin region where there is a fraction in the bottom of the ocean, in the ocean. And here is the fraction, the fissure, and we collect 10 points around the fissure, and each other points distance between 250 meters and 500 meters around these fissures. And there are very interesting results, for example, when we're looking for genes that are related to oil degradation, they are more abundant in the region of the fracture, here in red, than, for example, in the control area, or 250 meters and 500 meters far from this fracture. It's also very interesting results. And also, when we looking for the genomes that we can recover from this analysis, there are, for example, many Desulfobacteriaceae and Desulfobulbaceae, and genes and organisms related that is already known that these genomes have the ability to degrade and can deal with this oil in the environment. Another work is that we performed here in the Guanabara Bay, using the sediments of the Guanabara Bay. And was interesting to note that we collected samples in 0 meters depth, 1, 2, 3, and 4 meters inside the sediments. And it's interesting to note that, for example, there are some genes related to sulfur metabolism that are more abundant in the deepest depth than in the lower depth. And not only this, but when we try to assemble genomes from this analysis, 2 things happen. First is that this segment is so diversity that we can recover only 2 genomes from this metagenomic analysis. But interesting, one of these recovery genome was an archaea genome, a Bachiarchaeota, that have the ability to deal with sulfur cycle. And the second one is a genome that belongs to the related to the acetobacteria, but we discovered that this new genome is possible, belongs to a new order inside here, inside the acidobacteria, and we call it the Candidatus guanabacteria. This is a new order, and this genome shows more than 60 differences in silico phenotype comparing to this closed related genomes found here and publicly available. And most of them, as you note here, is the UBA. UBA is the genome recovered from a metagenomic analysis. This is also another work that we analyzed, algal mats, microbial mats from the Araruama Lagoon region system. Here is the location. And when we're looking for only to metagenomic data, we can identify many differences between the samples. They're very homogeneous around the time and between the samples of location. But interesting, we was able also to recover 2 genomes from this data. And one of these genomes is a new species that we call it Thialocarpa araruensis. And another genome is the BIN9, belong to the Saprospiraceae family. And when we're looking for the genomic metrics comparing to its closely related genomes, this is one, we notice that this is a new genus, and we call it Ararubacter turfi. And here, as this genome was recovered from a high salinity ambient, and there are many genes related to allotollerance recovered in these genomes. And there is a list of the genomes and the absence or presence of allotollerance genes related to these MAGs recovery from these samples. I'm almost finished. But this is also an interesting work that we sequence the metagenomic and we sequence the water from the Doce River. Immediately after the disaster of the rupture of the Fundão Dam, and we draw an experimental design that we collected immediately after the rupture of the dam, 6 months and 10 months, and there are here 2 control areas. And we notice that immediately after the rupture of the dam river, there are increase in genes related to iron acquisition and metabolism. And when we assemble metagenomic, when we assemble and we try to recover genome from this metagenomic data, interesting, we found a considerable number of genes related to Fa, to ferro, iron genes here. And we also can address this methodology not only specific to bacteria, but we can also work with viruses. And this is a work that we published years ago, working with the continuum of the Amazon River and the ocean continuum, and specifically analyze the plume of the Amazon rivers also. And when this data, with this metagenomic data, were assembled, we recovered almost 30,000 scaffolds. Of that, 15 are new completed viral genomes. And this was an interesting work because there are two peoples that one is collected inside the river, and other was collected in the ocean, and the salinity varies a lot. Here is almost a point that we call it is have the biophysical characteristic of seawater. And in here, these points here, the 10 and the 11, is a transition point and has a characteristic of the river and sometimes of the sea. And when we looking for what is the possible host of these genomes, we looked at, for example, we found that the points in the river shows a different possible host, and so we found many eukaryotic viruses in the sample from the river. And when we're looking for the samples in the sea, we look in many Prochlorococcus phages and Synechococcus phages. And this is the abundance of the 30,000 scaffold that we found in this analysis. And we can notice that, for example, there are a differential expression, a differential abundance in some virons, in some viruses in the ocean when we compare to the river samples. And this is this is the viruses that we can looking for and found in this analysis. And these viruses are interesting because abundance is almost a bioindicator. For example, this first virus is here, which is a very small one, but it's a complete viruses. Its presence in the river, its presence is an indicator that we are working with a sample from the river. And this blue one here and this one here are viruses that it's almost a bioindicator of sea samples, for example. And this is interesting because you can apply this methodology not only to bacteria, but also to viruses. And this work is very interesting because we show the potential of this technique to access and recovery viral genomes from this metagenomic data. And to finish, this is my last example. Yesterday, Cristiane asked to Matt and concerning the evolution and speciation of some bacteria in the world or in the sea, and this is a work that we published 2 years ago, and we looking and analyzing the Prochlorococcus collective. And we propose a new classification of this Prochlorococcus because, for example, the genome size and the GC range of these genomes is very different and varies a lot. And here we use it not only the cultivatable strains, but only genomes from single cells spread around the world. And in this specific case, there are some Prochlorococcus-like genome recovery from metagenomic assembly, but in this specific case, we don't use because we work it only with almost completed genomes that have the completeness of more than 90% of completeness. And when we looking for the characteristic of these genomes, here in green is a genera, a genus that we call it Euricolium. It's characterized by a lower GC content and a small size of the T genomes, contrasting, for example, with the Ptaumococcus here, that is a Prochlorococcus-like genome that shows a very big GC content, near of 50, and the GC and the genome size around 2.5 megabases. And here is the phylogenetic tree using 250 core proteins. And as we can see here, there is a monophyly of these proposed genomes. And interesting, when we looking for the Prochlorococcus genome recovered from this work of the Donovan Parks and the Phil Hugenholtz group, they were able to recover 8 Prochlorococcus mags. However, the GC content and the genome size of most of these recovered genomes shows a very high GC content and shows genomes bigger than 2 megabases. And so when you're looking for this data, we not analyze it itself because the completeness was lower than 90%, and so we not use it. But at the time when we were looking for these genomes, this genome has the characteristic not of a Prochlorococcus itself, but of the new genera that we call it, that we classified as Ptaumococcus. So yeah, that's it. Thank you very much for your time, and I'm now open for questions and doubts. Thank you very much for the invitation again.
Thank you very much, Diogo, for the very nice presentation. We really appreciate. And we are right on time, so we still have a few minutes for a couple of questions. Juliana, do you have some? And then I will ask perhaps one on one.
Yes, we do have a few questions on the chat. So the first one, Dr. Tschoeke, is what are the most effective ways to avoid contamination in mags?
Most effective ways to avoid contamination in mags. Very good question. It's not so easy to answer because, for example, the most effective ways is, for example, when you assemble the genomes, it's there are some parameters that you can use, for example, as in the spades, use the meta spades parameters, for example, and work with high k-mer number of the assemble. For example, use a k-mer size of more than 100, for example. And because when you use higher k-mer number, you decrease the probability to join reads that doesn't have the same k-mer profiles. And this is one way to avoid this contamination. But of course, there are not specifically the most effective ways. And of course, we need to analyze the quality of the mags before we get these genomes. For example, use the Tschoeke. Yesterday I was doing this presentation. And for example, the Tschoeke program, the paper of Tschoeke, has almost 5,000 citations since 2014. So it's a very useful program to show us this contamination or not.
Oh, indeed. Wow, great. I think we do have another question. Yes. So Carla is asking, what are the biggest challenges in when working with mags?
Okay, biggest challenge. I guess the biggest challenge is not necessarily the tools itself, but is the computational structure to work with mags, for example, because we cannot assemble a genome, for example, of 10 giga in our personal computer or in a regular desktop. You will need, for example, to use a server of that have more than, for example, 255 giga of memory, because most of the assembles use the Bruegel approach, and this approach needs a computer with with a high amount of memory. And so I guess that the biggest challenge is not specifically, of course, there are biological challenges, for example, to give the meaning of the genome that you recover, but a computational, I guess that the computational challenge is worse than, for example, the biological. Of course, there are some genomes that you don't know nothing. Only, for example, that you know that's a bacteria or an archaea point. But we can annotate this genome. But until you get there, you will need a good computational structure to do this analysis, I guess.
Great, thank you very much. I do have a question from myself as well, if you don't mind. I was wondering about the oil spill work. So did you find any relevant genes related to oil degradation during the functional adaptation of the metagenomes?
Yes. I show very, very quickly, but yes. And when the first or the second one that you mean?
The first one.
The first one, yes. When we compare because the water was incubated through 60 days, almost 60 days. It's a case control experiment. We have only the water with the organism itself and water plus oil. And in the sample that we have water plus oil, there was an increase of the abundance of genes related to oil degradation. So yes, we noted that, and this difference is statistically significant between these two this control and case and control analysis. There are an increase in this abundance of these genes, even not only in the genes, but in the organisms related to oil degradation.
Great, thank you very much, Dr. Zhou.
One last question, Diogo, so you can, I know that's a little bit off bioinformatics field itself, but of course you are and your group maybe are thinking about how we go from mags to applications, right? So you detect the genes related to a process, you detect organisms that might be related to a process. How go from your data to real applications? For instance, not only, I think I'm just using as a markers for an environmental process, or you're going to use the genes, cluster of genes in hosts, or using the actual organisms to in bioremediation process and so on.
Yeah, very interesting questions. Yeah, and it depends, for example, but in our group, mostly looking for this genome, analyze these genomes, and try to identify, for example, interesting genes related to oil degradation or antibiotic production, for example. But go further in this analysis, for example, it's not so easy and demands time. And this works, for example, most of these works were not so old, and so we didn't have the time to go further in this analysis once we get this genomic data. But most of this genomic that we assemble from these metagenomes are publicly available, and we hope that, for example, other group also can help us to go further in this analysis, I guess, because it's not so easy, for example, to get a full operon, for example, and clone in a bacteria and make the bacteria express, for example, antibiotic resistance. So we have some collaborations and try to go further in this analysis.
Okay, great. Thank you so much again, Diogo, for the fantastic talk. We really appreciate your presence here and helping us with the workshop. And we are out of time, and the next speakers are already setting his talk.
Thank you.
Again, thank you so much, and I'll see you next time.
Yeah, okay. Thank you very much also for the invitation and for this delightful workshop also. Thank you.
Thank you. Hello, everybody.
Hi.
Hi.
Thank you so much for accepting the invitation. Hi, Karthik. Thank you.
Gauro.
Zhichao, Patricia, and Juliana will introduce your group, Karthik, and then you can introduce Patricia and Zhichao for us.
Yep.
Okay, so go ahead, Juliana.
Great. Hello, everybody. So welcome to our anchor event. We are very pleased to introduce today's speaker, Dr. Karthik Anantaraman. He's an assistant professor in the Department of Bacteriology at the University of Wisconsin-Madison. He got his PhD from the University of Michigan in 2014, studying deep sea hydrothermal vent microbiology using multi-omic approaches. His interdisciplinary research focuses on understanding the cycling of sulfur and nutrients with a strong emphasis on the microbial and viral processes that transform these cycles in environmental and human systems. His lab is at the forefront of developing bioinformatics and statistical approaches to investigate microbiomes, viromes, metabolism, and community interactions. And today, he will talk to us about using metagenomics to study microbial biogeochemical interactions and viruses. Dr. Karthik, it's a pleasure to have you here, Patricia and Zhichao. So I'll give you the word. Thank you very much for accepting this invitation.
Thank you, Juliana. And thank you, Professor Ricardo, for the invitation. The way we'll do this is I'll start off and I'll provide an introduction about my laboratory, the kinds of research we do. I know at the end of the day, Anchor as an organization is associated with marine work and marine research. So our lab actually has huge interest in marine microbiology. So I thought the first 10 minutes, let me give you a little snippet of the different kinds of research our lab does, the motivations we have. I'll show you some of our pictures from fieldwork, and then I'll hand it over to Patricia and Zhichao here, who will talk about two specific tools that we have developed and are very useful in helping us understand microbial metabolism and biogeochemistry and viruses, primarily from metagenomic data. So we can start off. Patricia, do you want to start sharing slides? I will also apologize in advance that just a little after 9, I have to leave, but Patricia and Zhichao will continue the presentation and also stay back to answer any questions that may arise at the end.
Sorry, I have a little bit of trouble to share my screen. Zhao, would you be able to take a look, please? I'm sorry about that.
You mean that I will share the screen, right?
Yes, I'm sorry. It says that I'm not able to. Okay, I can see the slide.
And Chao, you have control?
Yes, I think it's on me now.
Okay. All right. So I'll just let you know when to move forward, and yep, you can go ahead. Okay, that's great. So my laboratory at the University of Wisconsin-Madison is what I think of as a microbiome and virome laboratory, and we really work at the interface of three different fields. So we work at the interface of biogeochemistry, microbial and viral systems ecology, and computational biology. And much of the research that happens in our lab, even though we do a lot of bioinformatics, there's two fundamental questions that really motivate us. The first question is trying to understand the roles of microbes and viruses in driving sulfur metabolism in environmental and human systems. And the second question is trying to trying to study viruses and then trying to use viruses to potentially modulate microbial communities. Next slide. So there's three major themes in our laboratory right now, and these themes are also associated with three kinds of field sites. When we started out, we primarily started out as a marine microbiology lab. We focused on studying hydrothermal vents and the deep oceans. And over here, we are interested in inorganic sulfur transformation. We've got field sites all over the globe. And you can see the field sites listed over here. So we've got field sites at East Pacific Rise, so that's off the coast of Costa Rica, at Guaymas, which is in Baja, Mexico, at the Southwest Indian Ridge, close to the Maldives, and at Lao Basin, which is between Tonga and Samoa. We have now expanded and diversified, and we have interest in freshwater microbiology as well, where we are studying inorganic and organic sulfur metabolism. We study both temperate as well as tropical lakes. So we've got field sites here in Wisconsin, as well as in Africa and Lake Tanganyika. And recently, we've also started studying the human microbiome, specifically organic sulfur and its association with diseases such as colorectal cancer and intestinal bowel diseases. So given our focus on marine systems, I thought I'll show you some of images as well as videos from our recent cruises. So most recently, we've gone on cruises to the East Pacific Rise and to Guaymas, Mexico. Zhou, can you go back to the video? Yep, can you play the video? So here is a video from Guaymas Basin where we have explored new hydrothermal vents for the first time. If you take your cursor, you can play the video. Take your cursor onto the image. Are you not able to play the image? Okay, that's fine. So we, unfortunately, we're not able to play this video. But yes, this video I was hoping to show you some images, or say a video from some of the newest hydrothermal vents that have been discovered in Guaymas Basin. But that's fine. We can move over. There are still some images that hopefully will show up. Here, I wanted to give you a sense of what it's like to go on cruises, to go on submarines to the bottom of the ocean and actually sample down there. So what you're looking at over here is how very precisely we can sample different parts of hydrothermal vents. So you're seeing hydrothermal vents emanating these fluids that are producing these black precipitates full of metals, and using a robotic arm, we can actually sample high temperature fluids and relatively low temperature as well, very, very precisely. On the right, you can see it's a schematic of a hydrothermal plume, and very precisely, we can take a robot or even the submarine and sample at different points in the hydrothermal vent. Next slide, Sean. We've of late started using in situ filtration. So that is, rather than bringing water all the way to the surface and filtering it on the deck, we can actually filter water in situ at depth at the bottom of the ocean. So what you're looking at over here is an instrument, a filtration device known as the super sampler. And we can load different filters over here. And the different colors you see on the filters are essentially precipitate. So the darker the color, it indicates that we've sampled a hydrothermal vent or a hydrothermal plume. Let me show you some pictures of submarines and robots. So here you're looking at Alvin, the submarine, the most famous submarine out there, which helped to discover the Titanic. It's quite a surreal experience to go down to the bottom of the ocean in Alvin. Here is another picture of Alvin, and I really wanted to show this because this really highlights what it's like to do research. So Alvin is a multi-million-dollar submarine, but we scientists use like milk crates and engineered devices that we've just built up in the lab on this particular submarine. This is an image of ROV Sebastian. So it's a remotely operated vehicle. This is again something that we've used very, very recently. So instead of a submarine, this is actually operated remotely from the ship. Here is the workhorse of the ocean. So this is called a CTD. So this is a device that can collect water as well as and it has a lot of sensors. So you can drop it off the ship, and it allows you to capture different physicochemical parameters of lakes or the oceans. And depending on where on the physical or chemical parameters that you see, you can choose to sample at a particular location. And here are some, fun and some not so fun images as I wrap up my introduction. We see a lot of animals down there at hydrothermal vents, even though, it's a very interesting environment with a lot of chemicals and metals. You see a lot of animals. So shown here is a crab, an octopus, there's fish, there's also tube worms. And I put up a picture of a table tennis table here because a ping pong is the favorite game on the ship. You can imagine as the ship is moving around, it's difficult to play ping pong or table tennis, and that's why it's quite popular. And I wanted to highlight this for people who can appreciate marine science. At the end of the day, no matter where we are, no matter where we are never far away from plastic pollution. And even at the bottom of the ocean, close to hydrothermal vents, we see a lot of plastic pollution, and this is especially saddening. But it's the reality of life today. Next. And I just wanted to show this last slide. I mean, this is a beautiful image from Guaymas Basin. So this is a hydrothermal vent that was observed for the first time. As you can imagine, it's a very large hydrothermal vent that's producing a lot of hydrothermal fluid. And what's highlighted over here in the inset rectangle is, if you observe very closely, you can see a face in there in the hydrothermal outputs of there, or hydrothermal discharge. You actually see the outline of a face. When you're on the ship for a long time, you're there for weeks and weeks and weeks. Often, you go through images, and this is the kind of stuff that you find in images. And with this, I'll hand it over to Zhichao and Patricia. We apologize that we couldn't play the video, but the link to the video is in the chat. So please feel free to watch the video. And Chao and Zhichao will take over from here and describe two specific bioinformatic tools that are great for microbiome and virome analysis. And as always, I'll be around for at least a little while, and I'm happy to answer questions later. So will Patricia and Zhichao. And at any point of time, even after this talk, if you ever wanted to get in touch with us, our emails will be at the end of the presentation. Please feel free to get in touch with us. And with that, I'll hand it over to Zhichao and Patricia.
Hi, very happy to join the Biotec workshop. As we all know that the number of microbiome and virome data has been growing rapidly in the recent decades, with various microbiome and virome programs covering almost all aspects of environment and life science research. Specifically, microbiome indicates all the collection of all the genetic material within the microbiota. The microbiota is actually the entire collection of microorganisms in a specific niche. And for virome, there are two specific resources of viral genomes. The first is the virome, which is specifically targeting on the viral fraction of the environmental samples. The second one is actually viral genomes that are reconstructed from the bulk metagenomes. From about 1 decade ago, we have the Human Microbiome Project, Terra Ocean Microbiome Project, and Earth Microbiome Project that have been initiated and have been ongoing until now. And they contain both the 16S RNA gene data sets, and also the metagenomics. And Earth Microbiome Project further contains the metabonomics. We have those various virome databases. Those, for example, the global oceans virome and the human virome databases, those are labeled in blue. It indicates that they are actually acquired from the virome, which is which are specifically targeting on the viral fraction. And the red labeled metagenomic gut viruses actually are originated from bulk metagenomes. The last one, the gut viral database, they are actually contain both the viral and the bulk metagenomes. Today, in this talk, me and Patricia will present two software that are invented in our lab. Both of them are published in Microbiome, the journal, in 2020 and 2022. We will introduce the purpose of the tools, the workflows. We will provide examples of the inputs and outputs, and we will also give some examples of application of these tools in real research. Then I will hand over to Patricia.
Great, thank you so much. So I will give a presentation on the first part, which is on metabolic. So first of all, metabolic is a tool that enables the metabolic and biogeochemical analyses of genomes in microbial communities. And this tool is a free and open source tool that's available on our GitHub. And one of the particularities of this tool is that it has many outputs that are user-friendly, for example, spreadsheets, and also data visualizations. So here on the diagram on the right, I have an example of what happens when you're taking samples from the environment and sequencing its data. So at the top, we have a microbial community that's composed of many different members. And when we do a DNA extraction, we can, for example, get a metagenome, which would be all the DNA from that sample. And there's many different technologies to generate sequencing data, for example, short read DNA sequencing or a long read DNA sequencing. And then we can also do RNA extractions to generate metatranscriptome data to assess gene expression. So the input that metabolic takes is genomes. So one of the first steps to get a genome is by doing metagenomic binning. There are different tools that are available online to do this. But after metagenomic binning and quality checking the genomes, we have metagenome assembled genome, or MAG, which is shown by the different circles at the top. So for one sample, we have many different MAGs. So now another way is if, for example, we're growing an isolate in the lab or a culture, we can sequence directly the whole genome of that and get an isolate genome. And either of those two options would work as an input for metabolic. And if, for example, in your own research, you're interested in looking at those public data sets, it can also take MAGs or genomes, for example, from NCBI or from published literature. It's specifically metabolic will assess carbon, nitrogen, sulfur, biogeochemical cycles. And it also has a couple of other metabolism, for example, iron cycling, arsenic cycling, and things like that. And in addition to focusing on these biogeochemical pathways, it also has detailed metabolic annotations for the whole genomes, for example, central carbon metabolism. Okay, next slide. So metabolic has two different versions, which once we install it, you have both metabolic G and metabolic C. So metabolic G is the genome version, so it will annotate the genome. And then metabolic C is the community level analysis. So that's, for example, if you have many different samples and you're trying to understand what are the taxa doing in all your different samples, then I would suggest using metabolic C. Now I will go a little bit more in detail on the workflow of each. So first of all, for metabolic G, all that it takes here in green shows your input file. So you can either provide the metagenome assembled genome files in a FASTA format. That can be a nucleotide, but if you will already have translated your DNA into proteins, then you can also provide the FAA files. So one of the first steps is to take these proteins and run a couple of searches using different publicly available databases, as well as our own developed metabolic HMM genes. So some database that we use are KEGG, IGRFAM, PFAM, and DBCAN, for example. In addition to that, we have the metabolic HMM, which are focusing on the carbon, nitrogen, and sulfur cycle. So once that's done, we have many different annotations. It will have a motif validation step. And the motif validation step is looking for the specific regions of the DNA to really ensure that we don't have false positives. And then it will process the data in a couple of different ways and generate visualization, shown here in red and pink at the bottom of the plot. So in red, you will see that it creates the sequence file for all your hits for genes. And it also creates in an Excel file the metabolic results. And it will also create some figures that I will talk about, which are in PDF format. Next slide. In comparison, metabolic Z is very similar, but in addition, one of the inputs is that you can provide it FASTQ files. So the FASTQ files are the sequencing reads data. And the sequencing reads data are going to be used for metagenomic mapping in order to determine the abundance of each of the organisms that you have previously input. So because of this, it creates a couple of other visualizations shown here at the bottom in pink, which I will go and talk about and give you some examples. But for example, it will have some functional network diagrams as well as sequential reaction diagrams. And in metabolic C, we have also developed a score called the metabolic weight score, which is a way to compare different samples and compare different functional networks across samples. Next slide. Here is an example of the different outputs that metabolic has. So depending on if you have metabolic C or metabolic G, it will create different types of files. However, the majority of the files are common to both. If you'd like more information on the specifics of that, you can go on our GitHub page. So I want to talk a little bit about the MW score. So the MW score is this metabolic weight score. And the purpose of it is to address the lack of quantitative and reproducible measures to represent the metabolic interactions in community. Because you might have seen in some papers, there's network diagrams, and network diagrams is also something that our program creates. But sequencing and all different samples in different environments, it's hard to compare them by just looking at them visually. So the purpose of the MW score is to have a number that we can compare between different samples for these reactions of interest that will be normalized. So in summary, kind of what it does is that you have many different functions and all your different taxas as the columns. And here they're color-coded in yellow and green and red. So a yellow is something that's not very represented in the community, and in red is something that's very represented in the community. So we can see here, for example, that there's this one genome. If we look at the column that's all red, it probably means that this one genome is very contributes to many of the different cycles, which are all the rows in this sample. And we can look at these numbers between different samples to compare them to each other. Next slide. So now I'm going to give you a couple of examples of the data visualization. Metabolic will have 4 different diagrams, the nutrient cycle diagrams, which are done at the organismal level and the community level, the sequential metabolic transformation diagrams, the functional networks, and the metabolic alluvial diagrams. These plots are generated automatically as part of the metabolic pipeline, but you can also run them individually. It's an R script if you are interested in generating only one of these after the pipeline has finished. Okay, so the first example is the nutrient cycle diagram at the individual level. So the purpose is to note the presence and absence of the pathways in individual genomes or across the community. So metabolic genome, will do the individual genomes, and metabolic C for the community will do both the genome level and the community level. So the cycles are the nitrogen cycle, shown here in the top right. You can see, for example, the different elements. Arrows shows the specific steps. And if an arrow is in red, it means that the pathway is present. And if it's in black, it means that the pathway is absent. And the threshold that we use by default is 75% of the pathway needs to be present for it to be shown in red. However, in metabolic, you can change the argument to be something more stringent, for example, 90% or 95% or something like that. Next slide. So the community level one is very similar to the previous figure. Here I'm showing only the sulfur cycle, but we have this for, once again, the other cycles as well. Now, the only difference is that you will see a line that says genome, and this is the count of genomes that have this pathway. So for example, here we have 48 genomes that have sulfide oxidation gene, and that will be out of all the genomes that you have used as an input. There's also something called coverage, which is based on the read mapping. So based on the reads in that sample, how many of the, what's the abundance of the organisms that are able to do this reaction. Now there's no colors in this diagram, so do not worry if you don't see any red arrows. You would rather look at the number of genomes and the coverage. Now the next one is a sequential metabolic transformation diagram. This one the purpose is to show how whole biogeochemical pathways are broken up and how many genomes are able to perform these reactions simultaneously. So to give you an example, this is for inorganic element cycling, for example, sulfur and nitrogen. So in the first box here, we have sulfur oxidation. So sulfur oxidation is a multi-step process that goes all the way from sulfide to elemental sulfur to sulfide to sulfate. And sometimes organisms are only able to do one of these steps, for example, in red here, and also underlined at the bottom on the x-axis here. Some organisms might only be able to do this blue step, and some organisms might be able to do this green step. Shown in blue here, and then in green here at the bottom on the x-axis. And you can see that the bar plot shows the amount of organisms that do this reaction and their abundance. However, there are organisms out there in the environment that are able to combine two steps together. For example, A and B in pink shows that this organism is able to do both the red step and the blue step together. And you can see here, as pointed in the pink arrow on the x-axis, that the organisms that are able to do the two of those steps are really less common than the ones who are able to do, for example, elemental sulfur oxidation. So this is another way that metabolic allows you to look at the reactions in your microbial community. So we also have functional networks. Functional networks, the purpose is to visualize the different pathways and how they're associated with different organisms. This is also one of the plots that Metabolic creates. And the way to read this plot is that each of these nodes, so each of these points, these circles here in black, shows the individual steps. They're all labeled with the name of the reaction. And the size of the nodes, so the size of those circles, is the node degree. So how many different lines are coming out of it. So if it's connected to more other pathways, the circle will be bigger. There's also edges, and edges is just another term for the lines that connect two different dots. And these lines are the organisms that are able to do two of those reactions. Because when we think about organisms in the environment, they're not only a nitrogen cycler, they might also rely on organic carbon. They're not only sulfur cycler, they might also rely on nitrogen. So these organisms connect two different pathways together, and that is what the lines are showing. The lines are going to be colored by the taxonomic identities. So as part of metabolic, we rely on the GTDB classification. So it's going to use the GTDB classification to here, for example, show the phyla that these organisms are from. We can also look at the thickness of the edges. The thickness of these lines are going to be the coverage of the abundance of these organisms. Next slide. And then finally, we have something called a metabolic alluvial plot. So the metabolic alluvial plot is, the purpose is to see how different groups of taxas are associated with different reactions, which are part of different categories. So to give you a concrete example of this, I have shown you an example here. On this first column here, we have the different names of the taxas. And they're kind of linked to a reaction. So we can see here that we have a lot of gamma proteobacteria in blue, and they're linked to, if we follow the lines to each reaction that it has, we can see what reaction it's able to do. And then all of these lines are connected to either its carbon, nitrogen, or other sulfur cycling. So if we have many samples, for example, we can create a plot like this for all of our samples, and we will be able to see that, for example, in one sample, we have a lot of gamma proteobacteria doing X reaction. In another sample, we might see a lot of actinobacteria doing Y reaction. So metabolic has been used in a number of papers already, and we are very excited to see people use our software. And it has been applied to many types of different environments. So for example, in biofilms in Yellowstone, in marine ecosystems to study specific phylum of bacteria, even in groundwater ecosystems, and so on. So in summary, Metabolic is an open source and free tool that hopes to facilitate the annotation of genomes, or MAGs, with a focus on genes involved in biogeochemical cycling. Specifically for this project, we added some specific annotation validation steps, for example, the motif finding, and as well as multiple ways to explore the output data, for example, through spreadsheet and data visualization. And we also developed this metabolic weight score, which is used as a way to compare between different communities and samples in a standardized and reproducible way. And finally, we showed that it has and can be applied to different environments. And we can also use metabolic whether we're interested in just one genome or a whole community of genomes. On this, I will let Zhichao continue to talk about our next tools.
Okay, I will introduce the second tool, which is named Vibrant. The full name of this software is Virus Identification by Iterative Annotation. This software can be used for identifying viruses, prokaryotic viruses from both metagenomic assemblies and metagenome assembled genomes. It was built for identifying double-strand DNA, single-strand DNA, and RNA viruses. This software can be used to retrieve viruses from both viral and bunker metagenomes. This means that it can be used to processing DNA sequencing datasets directly from the viral particles that are separated from the cellular components or from the whole metagenomic sequencing datasets. The latter one can be very useful due to that it can leverage of the large amount of datasets that are not datasets of metagenomes that are not specifically intended for virus research, but they are deposited in the database previously, and we can take advantage to identify more diversities of viruses. In this software, we use the neural network machine learning approach to separate viruses from the non-viruses in the mixed metagenomic samples. And we can also extract pro-viral region from bacterial and archaeal genomes. And also, after we determine the viral scaffolds and the viral genomes, we can estimate the viral genome completely. We use the quantity of the predicted viral sequences as a proxy, relative proxy, to estimate the completeness. So it will be divided into 4 categories. The circular, which is also meaning the complete viral genome, high quality, medium quality, and low quality. We use the annotation matrix of both the nucleotide replication proteins and the viral hallmark proteins to determine the quality. The nucleotide replication proteins contain proteins that are related to nucleotide replication and relevant mechanisms. For example, the nucleases, the polymerases, and the DNA and RNA binding proteins. The latter, the viral hallmark proteins contain the proteins that are specific for viruses and are used for productive infection, such as the structural proteins, the terminase, the viral hornin and lysine proteins. So the vibrant can provide complete annotation information for the functional characterization of the viral community. As we all know that phages can encode the auxiliary metabolic genes, AM genes, to either augment or redirect the metabolism of the host microbes. It can change the whole, it can contribute a significant part of the whole framework of the biogeochemistry mediated by microbes. So this is a classical example of how the cyanophages infect the host cell. The viral encoded two genes, which are named PSBA and PSBD. Those two genes are classical viral encoded AMGs to enhance or to augment the photosynthetic activity of the host. So the viruses can take advantage of the energies and the substrates that are generated by the host cell for their replication. So to classify the AMG from the metagenome and also from the virome, the vibrant can assign them into specific metabolic pathways or even broader categories based on the annotation given by the CAG database. So this can help to characterize the viral community function in a very comprehensive and scalable manner. As we know that the viral genome can sometimes integrated into the host chromosome to become a pro-virus. And if the host is a bacteria, then it is called a prophage. Prophage can replicate itself passively along with the replication of the host cell. It can also enter into a lytic life cycle when it is activated and performs productive infection. It can happen when the environmental under specific conditions, for example, the host environment condition changes, or if the host cell is under some pressure, the prophage will enter into the lytic life cycle. Then we in the vibrant, we implemented this function. The vibrant can actually identify the prophage, can excise the prophage from the host scaffolds. It uses a sliding window approach to find the regions of the prophages. It is actually based on the V-score, which is a metric to reflect the viral-like signature of the proteins in the region. So starting from the boundary of the viral-like region, starting from the boundary between the viral-like region and the low V-score region, which is likely to be the host bacterial, host microbial scaffold, it will find the first viral-like annotation and started from there to cut and give the precise pro-virus region. As for example, at the right-hand side, there is a yellow region containing 4 proteins. But actually, those are hypothetic proteins that are not so confident to be assigned to either viral proteins or non-viral proteins. So it uses a very prudent method to only identify the first viral-like protein. So it will actually provide a more accurate region of prophage comparing to the other software. In terms of the workflow of the vibrant, it contains the following 4 steps. Firstly, it will annotate the input sequences. It will firstly remove the short sequences if the sequence is less than 1 kbp or the annotated ORFs are less than 4 proteins. Then it will use both the CAG HMMs and the PFAM HMMs to annotate the proteins. And also, the proviruses will be split from the host scaffolds by a cycling window method, as I mentioned above. I mentioned previously, and then it will do the first round of filtering. It will eliminate about 75 to 85% of the non-viral scaffolds in this step. And then in the next step, it will use the walk HMMs to further annotate the proteins. And then the rough excised prophages will be further excised based on the annotation provided by the VOG HMMs. Then it will generate a matrix that could be used for the neural network machine learning algorithm. Then it will use the machine learning algorithm to precisely separate viral sequences from the non-viral sequences. So, and it will also excise the pro-virus regions based on the annotation. And then we use, together with the machine learning step, we introduce the automated creation step, which can either verify or either change the predictions based on the neural network machine learning prediction. And this step will significantly increase the accuracy rate and also increase the recovery rate. And finally, in the step 4, Vibrant will do the annotation for both the auxiliary metabolic genes and also the other annotations for the virus proteins. For example, the nucleotide replication proteins, the viral hallmark proteins, and then it will do the genome completeness estimation. And finally, it will combine the results, which are mainly in the tablet formats, and generate visualized figures. In terms of the machine learning step of Vibrant, we used about 27 metrics. 3 of them are V-scores. We introduced the V-score, and V-score actually determines the viral-like nature of the annotation. We used it to describe whether a protein that is annotated by the three HMM databases, whether they are likely to be virus-related HMM or whether they are likely to be non-virus-related HMM. So the sum of the V-score based on the 3 HMM databases is actually a very good metrics to indicate the probability of a scaffold to be either viral-like scaffold or non-viral-like scaffold. Based on this algorithm, we do the plotting, use benchmark the data sets. We can distinguish the virus, plasmid, and bacteria and archaeal sequences based on the matrix. And it turns out that the recall rate is better or equal, has better or equal performance comparing to the other software. And at the same time, we to further improve our software, we decided to add two metrics. The first one is the gene density, and the second one is the TA36 RBS motif. Those two metrics will be added in the future version to see if the addition of those two could increase the recall rate, the recovery rate, or the accuracy rate. Then, in terms of the outputs of the vibrant, we have all the files documents stored in a folder named the vibrant underscore and your input file name. The first part of the result contains the FAST file, the GenBank file, and list files for the identified viruses. The second part actually contains the annotation, metabolic, and summary information of the identified viruses. And the third part contains the two folders, which contain the raw HMM results, the HMM annotation tables. The fourth part, we contained all the visualized figures. In the last part, we have two log files. The first one is for the annotation summary. The second one is for the whole running process. So we used them in many environment applications. They could be applied in many natural environments and human body places, and the man-made facility. For example, the wastewater treatment plant facility. And they have very good performance in identifying the auxiliary metabolic genes, because auxiliary metabolic genes has been the hotspots of research to indicate the important biogeochemical functions that are mediated by the viruses. So this is the summary. The vibrant can be used for both virum, the specific for specific viruses isolation, and also the bulk metagenomic datasets. The variant can uses neural networks and also use newly developed V-score metric to achieve high recovery rate to identify both the lytic viruses and the integrated proviruses. Variant actually highlight the virus encoded auxiliary metabolic genes and pathways, and the results are very user-friendly, and the result can be used to evaluate the whole viral community function. So, bioinformatic tools are very useful, and the metabolic software can help us to study the roles of bacteria in the biogeochemical cycling, and the vibrant can help us to identify various scaffolds from the metagenomic data. And we also have two additional software. The first one is the The first one is the Vira, which can perform binding of the identified virus scaffolds into the virus genome. The second one is the Propagate, which is a software that can help us to estimate the activity of prophages in the microbial genomes. Those two software either in review, the paper are either in reviewing or they are published in bioRxiv. The software are deposited in our GitHub repository, so maybe you can get into the GitHub page of our lab and take a deep diving into our software. So last one, I would like to express my gratitude to all the members in our lab and all the funding providers. We would like to also express our gratitude to the collaborators and the function agencies that support our researches. We would like to take some questions if we have some more time left.
Sorry guys, I was kicked out from the room for 3 seconds. Can you hear me? Okay. So I'm back.
Hello, can you hear me? Great.
We have time for...
Yes, we have a few questions for you. So thank you very much first for the amazing presentation. Everybody really loved. The chat is going crazy. So the first question for you is, what are the most effective ways to avoid contamination in eggs?
This is from the last...
Last one, yes, this is from the last one. Just a second.
Next one, please.
Yeah. Okay. So in the graphic of functional networks and metabolic alluvial plot, is there a way to filter specific functions? For example, only show sulfate oxidation or acetate oxidation?
At the moment in the automated, oops, can you hear me?
Yes.
Okay, hi. Thank you for your question. At the moment, the automatic version of metabolic, you cannot filter by the specific reactions. However, the R code is publicly available on our GitHub. And so we would only need to add a step where we filter the table to select sulfur oxidation, for example, or sulfur category. And if you need any help with that, feel free to email me and I would be happy to incorporate that as a function. Another way that you can explore specific function is by looking at the Excel spreadsheet. So in the Excel spreadsheet, we will have all the annotations organized by categories. And so you can filter that by the reactions of interest. Thank you.
Great, thank you very much, Patricia. We have another one. Is it possible by bioinformatics to distinguish a prophage from a free phage particle? How does it work? Is that a window the software uses to scan the possible connections between the host and the virus sequences?
Normally, a prophage will be, in our software, a prophage will be identified from the host scaffold, which means that the scaffold will contain the low V-score regions and high V-score regions. The high V-score regions will be identified as prophage. So for a free phage scaffold, sometimes due to the assembling process, the bias of the or limitation of the assembling process, the prophage region could break up from the host scaffolds. And based on that, we could, if that is the situation, we could not actually identify it or separate them apart. But sometimes the integrated prophage will contain integrates in their genome. So that is the single node. And for the second question, in our software, we didn't implement such step function in our software. But we know that there are other pipelines or methods that are invented by other groups to identify or to study the connections between the viruses and hosts. If you are interested in the connections between viruses and the hosts, you can use the approaches and methods provided by the other group. I know there are some papers related to this field. Okay.
Perfect. Thank you very much, Zhichao. What about archaean viruses? Do they have quite distinct genome signatures? Can you pick up these viruses by using Vibrant? Is there any limitations to it?
We have actually Vibrant can work on both bacterial genome and archaeal genome. We don't pay specific interests or emphasizes to the differences between the bacterial archaeal genome. So maybe that will be some topic of other researches to see if the archaeal viruses have distinctive signatures that separate them apart from the bacterial viruses. And that is very interesting direction that I think.
Patricia, I have a question, if I may. I made these questions for other speakers since you are being followed by many students, undergrads all over the place, and the work of these tools. And one question that comes very frequent and I'm repeating right now and asking for your thoughts about it. For instance, when you use different pipelines, or even if you use variations of the pipeline, you have different outputs. It's like different outputs. For instance, like few genes in a pathway or one gene in a pathway, and the question remains: is this real? Is this pathway is present or absent? Even if you use a cutoff of 75% to call it present or absent, and you may maybe the organism lacks like one or two key enzymes, so most of the other enzymes are present, but the organism are not able to metabolize that particular substrate, for instance. So however, about that and be confident when you say that that particular pathway is functioning and will have like breaking lacks or on the metabolic pathway.
Yes, absolutely. I think that's a great question. And that's something that comes up in metagenomic research because the metagenome is only going to give you the potential, even if we had a perfect ability to identify all the genes, the metagenome is as only a metabolic potential of what it could be doing. And there's multiple steps before what it actually is doing in the environment. So that's the first caveat. And then the second one regarding yes, the issue with many different tools have many different genes and different ways to identify the pathways. Well, there's a couple of main ones that I think people do. It would definitely be to look at the key genes in the pathway, and if that key gene is not present in the pathway, even though it's like 80% complete, but that key gene is not there, then the pathway is either the something in the assembly was wrong, the genome completeness is maybe missing, or there's something going on. So it's not like 100% perfect. Other ways I would also suggest is to validate the results between different tools and try to look at the actual scaffolds, because sometimes on the scaffolds we'll have, like, if it's an operon of gene and you like see two important one is hypothetical, one is something else, and we don't know what this hypothetical one is. We can have hints on what it is. And so I always suggest that even though we have all these bioinformatics tools that give you an automated way to facilitate this research, it's always good to actually look at the scaffolds and the genes to double check that information to get more confidence in the interpretation.
That's an excellent piece of advice. Thank you very much, Patricia. Okay, so we have another question. So can you identify new viruses sequences without a database bias?
Because our vibrant is not based on the sequence alignment method. It is based on the protein annotation result by HMM search. The HMM search can actually identify distantly related proteins. So this will enlarge the strength of the database, actually, because you have a database of all the all the viral-like HMMs, and you take advantage of the power of the HMM search to search the distantly related proteins. So this has significantly increased our limitation of the database. So it can have higher recovery rate comparing to the software that currently used in many papers. For example, the very real sorter, maybe real sorter 2, maybe.
Perfect.
Yes.
Thank you very much, Zhichao. So another one, does metabolic provides information on arsenic transformation genes?
Yes, so we have arsenate reduction genes and arsenite oxidation gene that are incorporated in that other nutrient cycle visualization. So there are some plots for that, as well as the Excel spreadsheet and with that information. And those HMMs are the custom HMMs for arsenic.
Perfect. Thank you very much. Do we have any other questions? Yes. Okay. So Osvaldo is asking, these results suggest that Vibrant has no evident environmental biases and identify viruses from a range of source environments. Are there differences on the viral community from these environments?
Yes, we mentioned in our paper that when we take a look at the viral community function that are implemented that are interpreted by the collection of auxiliary metabolic genes, we will see a clear differentiation between the man-made facility viral genomes and the natural environment genomes. If you plot a PKA diagram, we will see that they are clearly separated, separately distributed in the diagram. Yes.
Amazing. Thank you very much.
We have time for one last one, maybe. Oh, yeah.
Yes, we do have another one. What are the limitations of metabolic software? And there are any other software that do the same as metabolic?
Yes, so first of all, for the limitations, I would say that because you can install metabolic genome or metabolic community, metabolic C, the space requirements for metabolic C is higher because it relies on the prior installation of GTDB TK, which is a tool that some of you may know for taxonomic assignment of metagenome assembled genome, and that database can be quite large. So if you can run metabolic G on its own, but if you want to run metabolic C with the taxonomic assignment and the mapping, then you might need more computational resource requirement. Also, I think we're always trying to improve how to install the software, because right now we can install it via a Conda environment, which is the one that I would suggest, because it has many different dependencies. So sometimes that can be a bit complicated, but we're very responsive on our GitHub if people have suggestions on how to make that easier, if people have skills for that. But right now, the best way would be through a Conda environment. And then there are other softwares that do genome annotations using different types of databases. I'm not sure that there's one that specifically focuses on carbon, nitrogen, and sulfur cycling, but some examples of other software that do genome annotation are, for example, DRAM or Meta Pathways or Pathway Tools, or some that I know of. Yeah, thank you for your question.
Thank you very much, Patricia and Zhichao. We do have a few more questions on the chat, but unfortunately, due to our time limitations, we cannot proceed on that. But if it's okay for you, maybe we can advise them to send through your emails. We do have a lot of compliments as well on the presentation. So thank you very much. That was a very exciting and very fascinating talk that you gave us today. So thank you so much for contributing to our anchor event. And we really hope you can be presented also in our next events. Thank you very much, Dr. Zhichao and Dr.
Very nice presentation. Thank you so much for accepting our invitation. I really appreciate your time and your help in our workshop. Thank you so much.
Thank you so much, and have a great rest of the event.
Yeah, thank you. We'll see you next time.
Hello, everybody. Welcome back. So this is our third and final lecture for the day, and I'm really glad to having here Dr. Andrei Steindorff. He has a lot of experience in biochemistry and bioinformatics, with emphasis on comparative genomics of fungi. Now he's a postdoctoral fellow at the Joint Genome Institute, founded by the Doe Lawrence Berkeley National Laboratory, working with assembly, annotation, and comparative analysis of fungal genomes. He has a lot of experience in the areas of yeasts, filamentous fungi, assembly and functional and structural annotation of genomes, comparative genomes, transcriptomics, proteomics, and systems biology. And today he will be talking about Mycocosm, tools for comparative genomics. Dr. Andrei, welcome to our anchor event. It's a pleasure to have you here today. So you have the word now. Thank you very much for accepting our invitation.
Of course, yeah. Thank you for the invitation. It's great to talk about Mycocosm and Mycocosm, and I expect to get more, users to our platform, and not even to use the website, but setting samples, and I'll explain better during my presentation. Share screen. Okay, can you guys see my presentation? I'm assuming you're seeing my presentation. So today I changed a little bit the title. So because our group, we don't work only with fungal, we're working with algal genomes also right now. So today we're going to talk about the science we do with fungal and algal genomes, and some of the tools we use to do this comparative genomics. So at first, I'll talk about a little bit the Giant Genome Institute. So here's a picture of our website. So we have like a lot of things happening here. So if you want to explore, see what we do, we have like several different scientific programs. Like for example, we have the plant program, metagenome program, DNA synthesis, secondary metabolites. My group, which is this fungal and algal program, the microbial program is focused in bacteria and archaea and other prokaryotes, and metabolomics program. So we have a big infrastructure to do sequencing. So we use Illumina, PacBio, Nanopore, but mostly Illumina and PacBio for the for the DNA synthesis. We have like a very nice gene synthesis, so we can do a lot of different things that involve gene synthesis. Mass spectrometer for metabolomics. You have like all the flavors of mass spec that you can think about, so you can do several different types of analysis. And recently we implemented this technique called DAPSeq, which is you can detect some transcription factors, and then you synthesize the transcription factors. And when you synthesize, you add this affinity tag where it binds to a magnetic bead. So then you can link it to your naked DNA, and then catch this marked transcription factors, and then sequence everything that was bound to this transcription factor. And after that, you get the peaks when you map into the genome. So you get these peaks of where this transcription factor is bound. So, and then you can detect the promoters of the genes, and then detect the motif of this transcription factors bind. So it's a very interesting analysis. It's very high demand right now, because it then combines some of the biochemistry and some of the sequencing, and we're providing this service also. And for that, we have this community science program. We have several different calls over the years. Sometimes it's annual, sometimes twice a year. So it depends on your project. You can see the rules for each one. So if you have genome, transcriptome, like metabolomics, everything that you can work with for algal, bacteria, fungal, plants. So you can just submit your proposal. We're going to evaluate. If it gets approved, we do like everything for free. You can do like sequencing. You just send us the samples, and then we do all the processing, the analysis, the bioinformatics, the creative portals, everything, so you can access your data afterwards. And after the data will be public, but you have some period of, I think it's 1 year to publish your the manuscript using this data, because since it's user facility is funded by the DOE, the Department of Energy of the United States. So you have to make this data public. But yeah, you can submit your proposal, and then you do everything for free and make it available to you first, and then for everyone. So Joint Genome Institute, at first, it was located here in the Bay Area in Walnut Creek. Here's a picture of the first building since the 90s, when it was created to sequence the human genome. And I believe it was sequenced like 3 chromosomes of the human genome at JGI. And then because it was like a conflict with NIH, because it was like human focused, and then we moved, DOE moved JGI to work with everything not human or animal, working with plants, bacterial, everything that involved in energy, to create energy. And in 2019, we moved to Berkeley, to the Berkeley Lab campus. So here's a brand new building. We're in the first 3 floors. No, actually the 4th floor is half is K-base and half is JGI. So we're located here right now. So the first 2 floors are only labs. So we do all the sequencing, all the metabolomics, gene synthesis, everything's done in these 2 floors. And the other 2 floors are most like bioinformatics, just cubicles, people working computers. So it's a very beautiful place. Here's a picture of the cafeteria at the Berkeley Lab, so you can see the whole Bay Area. Here's the Golden Gate Bridge. So San Francisco is here. So in Berkeley, down here, so it's a very nice place. You can see like here's UC Berkeley. So this is a zoom the picture. So it's a very nice place to work. So today I'm going to talk about 2 things. One is science, what we do, like some examples of publications we did using all the infrastructure present at JGI, and some tools that we use for comparative genomics. And I'll show Mycocosm, which is focused on fungal genomics, and Phycocosm, which is focused in algal genomics. So here's my group. We have the Igor Grigoriev, which is the head of fungal and algal program, and other Igor, the Shabalov, he's the lead software developer, because we have all the portals, all the infrastructure, databases, everything that we have to keep on track. So we required a lot of this software developers. All these guys here, they're software developers. And we have some data scientists and postdocs in our group. So we have actually now we have 2 postdocs that's starting in our group. So we have like a big thing, a lot of fronts to explore. And JGI, so it started in the first, in 2004, we published the first Basidiomyces genome, was the Phanerochaetic chrysosporum. So it was the first one in 2004. In 2022, we have more than 2,000 genomes already available at Mycocosm. So here we have the front page is this phylogenetic tree where we have all the branches. So everything here is clickable. I can show you later how to do this. So you can explore, see like phylogenetic tree and some other information about all these groups. And the idea, because DOE is focused on energy, so these first projects were to find fungi that could degrade by plant cell walls, like plant biomass, and to degrade this plant biomass and then release fermentable sugars to produce biofuels. Was the first idea. And then the fungi that does it like really well, this wood decay fungi. So and you have these 2 known strategies for this wood decay fungi, like the brown rot and white rot, where the main difference is that brown rot, they can't degrade lignin, and white rot they can. So that's why they put this 2 differences, because white rot, it's kind of, when they degrade the wood, it's kind of the final product is kind of white because there's no lignin anymore. So that's why they call this name. So this is a big difference. But Robert Riley, he published this paper in 2014 at PNAS, where he showed that this is not a dichotomy, like brown rot and white rot. You have some other guys that is kind of in between. So he proposed that this is a continuum, is not like a binary state where they do or do not. So, and then he called it like gray rot. So this was a very interesting paper to read about this methods of degrading wood. And since the first genome of this Phanerochaetic chrysosporum, and then after in 2012, he published this paper in Science with 16 genomes of this Basidiomyces. And then in 2014, this proposal, this difference between brown rot and white rot and brown rot was a continuum. And nowadays you have like more than 150 genomes of this wood degraders fungi. So this is a thing that we try to explore more and get more samples and understand how this process works. And other front to get to this biomass degradation is this Neocalimycegomycetes, which is this gut fungi. Is a very powerful lignose degraders. So here I'm showing the tree. There are these early diverging fungi, and they are very tricky to work, even like for sampling and to work with the genomes. Just as an example, I remember the first time I've been here at JGI. I was like when they were training me how to do to import data, like external data to JGI, to Mycocosm. I was importing this Orpinomyces sp, and I remember seeing this genome. It had more scaffolds than genes. It was like a ridiculous thing. It was very hard to do because you had like most of the scaffolds had just one gene or no gene. So it was very weird because it's very tricky because it's AT rich. So the GC content is like 17%. I believe this one was like 15%. So it's a very weird when you open the file, just see ATs. It's kind of something very repetitive, and they're large, so they're the worst case scenario for fungi. So especially if you use short reads to do the sequencing, it's very hard to get a good genome from that. So the solution was using PacBio, because at first we were actually did this first genomes. I talked about this Phanerochaetes chrysospora in 2004 until 2008, 2010. Most of these genomes were using Sanger sequencing, so it was very hard to do. We had to develop like assemblers in-house to assemble this genome. So it was so very hard. That's why you get like these Nature papers from one genome. But and then the technology was evolving, and then we had Illumina. Was like for a little time we used 454, but I think wasn't that successful. And then implemented Illumina. But for this complicated genomes, Illumina was not working. So then when we had like PacBio, then it solved some of these problems. Because this low GC, like high repetitive, is very repetitive because it's, we have only 2 nucleotides, most of the genome, so it's very easy to get repetitive. So, and for example, this Pyromyces sp here, the genome with like 40 megabases with like 8,000 contigs. And when we sequence with PacBio, we got like 56 megabases with 232 contigs, was way better than the Illumina. Here showing like a picture of this largest contig, 2.5 megabases. You see clearly that how fragmented this Illumina genome was. And PacBio was good because it covered some of these more repetitive sequences here that Illumina wasn't capable to do. So it's a very good method. But PacBio is good, but it's not the silver bullet. It's still not perfect. Sometimes it's very hard to get good data because the problem is that you need high quality DNA. It doesn't matter the method you use. If you don't have high quality DNA, it doesn't matter. The genome will be bad. So that's another thing that has to be carefully addressed when you're trying to sequence a genome. But overall, it's very good. And the interesting part about this Neocalimyces is this cellulosome assembly. It was the first one described in fungi. The cellulosome is this structure that has like several different types of protein. When they assemble to do a purpose like degrade cellulose. So it's a multi-domain structure protein. So it's very interesting because it's very effective degrading cellulose, and they're specifically this cellulosome. They're Neocalimycegota specific, so they're only present in this clade, and a lot of these genes present in this cellulosome are actually horizontal gene transfer from bacteria, because when they live in the gut, they co-evolve with a lot of these bacteria, so they have this contact, so they got the transfer of some of these genes. That's why they are so good in degrading cellulose. So another bonus when you do like PacBio sequencing is the methylation data. So here's a paper we published in 2020, I guess, or 2019, where we found that in specifically these early diverging fungi, when you do PacBio sequencing, you find a lot of these 6MA levels. This early diverging 6MA is like the adenine is methylated in the carbon 6. So it's a very different type of methylation. So as you can see here, in other fungi is pretty much not found, but in early diverging, we find a lot of them, and not in other model organisms, not so frequent as in early diverging fungi. So what it does, we see these islands of the 6MA, like in the promoter region of the genes in this early diverging. And what it does, it has a positive impact on gene expression, in opposite of cysteine methylation, where you see like it blocks the expression. In this case, it activates the expression. So it's a very nice paper. So if you want to take a look at this data, I strongly suggest you. So I think today they can detect other types of methylation. Is not so accurate, but the sequencing can, because actually it's a problem with the sequencing, but we kind of figured out why it's a problem. Because when you do PacBio sequencing, you have like, it reads nucleotide by nucleotide with a certain time. And when you have like a delay in the read of this next nucleotide, and then when the delay is caused because the base is methylated. So, and you have like different delays for different types of methylation. So, and some of them are more noisy, but some of them are more clear. Like this 6MA is a good example of where you could use this problem with the sequencing actually to find new information. And now Nanopore, they are also doing that. Actually, we have a big project trying to explore different types of methylation, different types of organism using Nanopore, but it's still kind of like noise is not so accurate. We're trying to improve the data, but it's so far I think it's very promising. And other things not necessarily related to degrading biomass is this. We use like single cell genomics to fill up some gaps in the phylogeny, because if you look, for example, in the Mycocosm tree, like 90% of the genomes are coming from like Dicaria, or just like probably all the fungi that I've heard about are from this clade here, and only 10% are from this early diverging fungi. And the reason for that is that most of the genomes are most of these fungi, they're very hard to cultivate. So if you can't cultivate, you can't extract DNA, and then you don't have a genome. And to do that, we developed this method to do like single cell genomes, because most of them are aquatic. So then to isolate the specific cells for this fungi, and then extracting DNA, and then doing amplification of this DNA, because you only have like one cell, so you need to amplify this DNA. So do several steps of PCR, try to amplify this, and then do the sequencing, sequence rDNA to detect the species, and then do like cell by cell doing the sequencing, try to get like good genomes. So we developed this workflow to sequence single cell fungi. And here you can see the results in this paper, the Nature Microbiology in 2018 from Stephen Art. So we see that they miss the same metabolic pathways here in red. So all these single cells that's hard to cultivate, they don't grow in like a medium, because they miss some of these metabolic pathways. So, and you see that most of them, they miss the same pathways, but we still find the new like protease and secondary metabolized proteins in this early diverging fungi. So here we're trying to fill up the gaps in the phylogeny, because most of these guys here are very hard to find. And other approach we use was this, because we have a lot of genomes that are plant pathogens, in case of these dotidiomycetes. So here, I'm sure like a paper we published in 2020 for this 101 dotidiomycetes genomes. So we could reclassify like 25 of these organisms, because they're very hard to do the phylogeny. So when you have the genome, it's easier to perform these phylogenies. And we use this machine learning method where we're using only like 6 gene clusters. We could classify plant pathogens and saprobes. So when you have the genome, you'll get the 6 gene clusters, and only based on this information, you can know if these guys are plant pathogens or only regular saprobes. and I think it's a very interesting like method. So it was published in Studies in Mycology in 2020. So if you also are interested in like plant pathogen, this is a very good paper. And this one is one of the oldest projects we have at JGI, is the 1,000 genome project, because when we started to sequence genomes, like everybody was thinking about like 1,000 genomes for everything. But here we're trying to sample different clades, not because getting 1,000 genomes from the same clade is very easy. For example, in Aspergillus, you already have like almost like 400 genomes only for Aspergillus, because they're, they have a big community. Like people are all the time like sampling different species, so they have a very active community. So it's easier to get more genomes. But what we're trying to do is getting 1,000 genomes where it's sampled from all the fungal tree. Is not only one specific clade, as you can see here for Staudidiomyces, because they're plant pathogens, they have a lot of them, and then like Sordiomyces, Garicomyces. So this is more common fungi. We have like a lot of these samples already. So what we're trying to do is getting new ones. So if you, for example, work with some of this fungi and you see that is not sequenced yet, for example, we have like some cases here, you can go to this page, this fungi 1,000 projects, and then you can nominate, for example, if, I have this in my lab, I'd like to sequence it, you can just come here, click, and then go through the process, and then you can apply to a CSP. In this case here, I think it's a fast track, because there's already a CSP approved. So it's easier, just send a sample, the DNA sample, the DNA and RNA, and you can sequence the genome. So I think this is a very cool tool that we have to fill up the gaps in the phylogeny. Yeah, so not only fungi, I think most of you, because you're marine biology, are interested in algal. So I'll talk a little bit about the algal program and what we're doing right now. It's a new program, so that's why we don't have like that much data, but we're trying to explore more this area, because as I told you, like for fungal, we have like started in 2004, and in like 18 years we have like more than 2,000 genomes right now. So the focus now at least in the fungal part, is working like with pan genomes, multiomics, like do like metabolic modeling, and try to address like some of these unknown genes, and maybe do gene synthesis to identify the function of this unknown gene. So we have a lot of these different ways to explore the data we already have. So we're getting new genomes, but I think we already have like most of that. These are all the common common species. We already have most of them. So we're trying to do other types of approaches. But for algae, we started the first project, I think it was around 2004, but algae is way more complicated, because it's something like more close to plants when you think about like the complexity of these genomes. And around like 2009, we had like 10 genomes. And now we have like around 100. Actually, I checked yesterday, we have like 153, I guess, genomes from algae, because I think because this one was before we created Mycocosm. These genomes were sequenced by Phytosome, which is the plant part of JGI. And then we created this portal specific for algal genomes. So it's very complicated, because as you know, like algae is very hard to get extended cultures. So usually either you get them, they're contaminated with other bacteria, sometimes endosymbionts like virals. It's a mess, and to get like good assemblies is also like complicated. So that's why I think it grow like in a slower pace than fungi, which is way more simple to cultivate, to do all the analysis. But we're trying to expand. Here we have like this clades in gray, is because we don't have any genomes from these clades yet. So we're trying to get new samples. So if you work with algae and you have like some of this interesting clades here, I think it's very easy to get approved a proposal to do like the sequencing and even other analysis. If you're interested like in biofuels, or even creating more biomass, or the contamination, whatever the you want to work with this, guys. I think it's a very interesting topic right now. And yeah, here just showing some of these genomes that we sequenced at JGI. So the first one of the Chlamydomonas. So this green algae clade here, but then we got other of this Haptophyta, like Cryptophyta. So we sequence a lot of these guys here. Some of them are just released. So we're trying to expand the phylogenetic tree in algae, really getting this new samples. And one topic that we're working right now is because in fungi, as you can see, we're trying to find fungi that could degrade biomass to get biofuels. So for algae, it's the opposite. We're trying to get algae that can accumulate, in this case, like lipids or all the molecules that can be used as biofuels. So one approach we're using is this. For example, these two strains, one Cenodesmus and Monorphidium. One is very good to grow in the summer, and the other one is very good to grow in the winter. So we're trying to improve these strains to produce more, to understand how they produce these lipids and what time of day, what type of, for example, salt concentration, other metrics. We're trying to understand how they was the best situation or best condition for them to produce this lipids. So we have this the genomes, and we have a lot of these transcriptomes data, and we're combining them with metabolomics to understand how these pathways are regulated, and then create these co-expression networks, and then combine this with lipidomics in this case, where you can address like the how many double bonds and so the different types of lipids that are produced by these strains in the different conditions, and what are the transcription factors that regulates this. And after that, you can engineer this these strains and then improve the production. So this is one project that's led by Sarah Calhoun. She's already published a paper in this topic. Now she's working with a different one where she's like looking at different salt concentrations, not only temperature. So it's a very cool area that you're working right now. So okay, so in algae we're starting now. We have other fronts also. For example, we're trying to improve metabolic modeling, especially in algae, because most of what we have information is coming from plants, but algae is like they're not necessarily like exactly the same as plants. So we're trying to improve this metabolic, getting like better ways to address metabolism, and then building new pathways, because most of these pathways are exclusive to plants. So, but algae, they have some different things happening there. So we're trying to improve this metabolic mapping. So we have like some other postdoc work in this front. So I think in the future we're gonna have like other very interesting things involved, implemented at Mycocosm and at Mycocosm also, because you're implementing both of them together, showing this metabolic mappings of these guys. So now I talked about the science we do at JGI. So now we're going to talk about the tools we have to do this comparative genomics. So we start to talk a little bit about the karyotic genome annotation pipeline. That's what we do pretty much in our group, because at least at JGI we have the groups are separate. For example, for assemblies, we have a specific group that work with only assemblies. So the expert in doing genome assemblies. So, and we do annotation. So here I focus more about the annotation, which is more complex to perform. So because eukaryotic genome annotation is harder in a way than bacteria, than prokaryotes, because prokaryotes pretty much only had to find the ORs there, and there's not much to curate. You have to do some curation, but it's not so complex. Here we need like a lot of massage in the data to get like the right gene models, because at least in our group, what we do here, the step by step, what we do like, you get the genome assembly, and then we do repeat masking, and then map the data. What I mean by map the data is what you give for support your gene predictions. For example, transcriptome is a type of support data, but you can use proteomics. So everything that have another proof that that gene exists, we use to support the annotation. And then perform the gene prediction after this mapping, and then the functional annotation, and then validation, where everybody like take a look in the genome and see if it's makes sense or not, and then release to the public. So for the gene prediction, we use these three types of predictions: the ab initio methods, which is pretty much only look at the genome, get the training based on like intron size, like the proportion of each nucleotide, all this, get all these metrics, and then train the predictor and look at the assembly and then tell you what is the gene. Starting like it has to start in ATG. These are the exons, introns, and the stop codon. So that's it. It's only looks at the assembly. So I put some examples here: the FGenes and GeneMark, two of these predictors. And then we have a transcript-based method where you need the EST. So this is coming from the transcriptome. So you use the raw transcriptome and also the assembled transcriptome. You usually use Trinity to assemble these transcriptomes. And then based on this data, we map against the genome. And then based on this mapping, you predict where the gene is. For example, this is an UTR. The gene starts here. You have a gap for intron. And then this stops here, and then the UTR. So then this is usually the most like accurate models, because the transcriptome is coming from your strain, so it's not relying on other people's work. So it's from your data. So usually it's more reliable. That's why you have to treat it very carefully to get like a good transcriptome assembly, and then to predict the correct models. And then you have the protein-based methods where you have to map proteins. Usually get proteins from GenBank or SwissPro, other my curated data sets. So we get this protein and then map the protein against your genome. And then based on the seeds of this, usually we use BLAST to get the seeds, and then we extend to get the gene model based on this other protein that was predicted for other genomes. So here's a more like overview of this, how this method works. So as I said before, the genome scaffolds. So these three parts here are done in parallel. So you mask the scaffolds using repeat scout to get like de novo repeats. So you see like what regions in your genome is like highly overrepresented. So we consider that like repeats in your genome. And then repeat masker, which uses rep base to get this known repetitive content. And then you combine them to mask your genome. And then map the RNAs from your contigs and from the RNA-seq to get these transcript-based gene models, and then the ab initio and protein-based. And then after that, we annotate functionally all the genes, because here, as you can see, you have like several different gene predictors. As far as I remember, last time I checked, the full pipeline we use, it has like 13 different gene predictors. So what we do, we predict genes using all the gene predictions in the whole genome. So usually have like thousands of gene models from each of these gene predictions. And then we annotate functionally all these genes using, for example, InterProScan, TMHMM, like SignalP, and BLAST against SwissPro, CAG, COG, proteins, all these different types of databases. And after that, we do the filtering. So this is the part that makes all the pipelines different, because you have a lot of methods published that does this type of annotation. For example, for fungal, we have the FunAnnotate. There's like FunGap, there's Maker. There's a lot of different programs that does the same thing, more or less. So, but the big difference on them, like usually they have like less gene modelers than us. We try to get like all the good ones at least and try to implement here. But then you have to select what is the best model for that loci. So that things gets like very complicated, because you have gene models that are ab initio, or they're transcript based, or they're protein based. So which one is the correct one? So here we have a heuristic where you have like different weights for different methods. So usually we give more weight to transcript-based gene models, and then protein-based, and then ab initio. So then we have this heuristic to select what is the best model. Because sometimes we see that other pipelines where they use evidence modeler, for example, is a program used to do this filtering, but it's a very simple filtering. You just give like a weight, and that's it. It doesn't look at any other metrics. So, but here we always like trying to improve this heuristic, where we looks at the gene size, if there's like UTR, the types of that domains or not. I have a lot of different things I've had to look at this gene to select what would be the best model for that loci. So after we filtered the best models, then you have this gene set where we call filter gene models. So these guys here are the best gene models for each loci. That's what you see, for example, in the portal, or when you download. That's the main one that we use. And then we do other types of post analysis like OrthoMCL or BLAST or other things using this data set. And for algae, we do like something like similar. What we have that there's some differences. For example, for repeat masker, for fungi we only use the fungal rep base, but for repeat masker we use for algae we use all the rep base because it's more complicated. We have a lot of different repetitive regions, so you try not to rely only on plants. For example, we're using everything. And the BLASTAC step here to get this seeds for the protein gene models, we use this MMETSP, which is a transcriptome database for algae, specifically for algae. So we added this other database to get more protein seeds. And the modeling, we have a more relaxed intron length cutoff, because in the fungi we use 2,000-3,000 base pairs for intron length, because it's very rare to find intros like longer than that. So we kind of restricted the introns, but for algae, it's not the case. You have to give it like a more relaxed cutoff. And also the filtering, we modified and we prioritize like completeness of the genome. So we have like Busco and Sigma genes. So we try to get the best set that represents more the completeness of this genome. But it's a constant modification. Sometimes you get like a new genome, which is completely different, which is common in algae because they're like in every place in the phylogenetic tree. So you get like a more complex genome, then you have to double check every step, what if it's not working, what you have to do to fix this. So it's a constant work in progress to annotate these algal genomes. And the final step, that's one thing usually people don't do, and we do, which is very important, is this manual validation. So for example, I annotate one genome and I send it to other like data scientists in our group to double check this annotation if it makes sense. So then you have like other eyes looking at your data, so it usually is more reliable. We have a lot of the steps. For example, one is the phylogenetic tree. So after you assemble the genome, annotate, and then you build the tree based on this gene set you get, and then you look at the tree and see if it makes sense. If the organism you're looking at, oh yeah, that's the position I was expecting to be. If it's not, you can talk to the PI of this project and, oh, I found this genome here. Does it make sense? Sometimes it does, because they're trying to reassign the phylogeny that organism. So it's very important to check the position here in the phylogeny. And also other like genome. Usually this part is not so important because the assemblies usually is done by the other group, and they check all these steps, this metrics here, like the size, N50, L50, repeats, ploidy. So because sometimes you get like genomes that there's diploid, so you have to be careful with some steps if it's high repetitive. So some things you have to be aware that you're dealing with this type of genome. And then you check the gene model sources. For example, what gene predictor got more genes in the filtered set. So usually we see like a balance, like more for transcript-based methods, and then for protein-based, and then for ab initio. So this balance is very important. Sometimes if you see way more genes from ab initio because something's wrong with your transcriptome, why they're not mapping, you have to check why this happened during the annotation. And then the protein length, this is also very important. I'm showing here like an example. We have the protein length like in bins and the number of proteins. Sometimes you see like all the genomes are kind of following the same trend, and then you see one guy that has like this way less or way more than expected. So what happened there? So it can be like the database you use had a lot of bad proteins. Sometimes it happens when you use NR without like any filtering, because NR like has everything that people submit. So sometimes there's some bad gene models, and then when you use that to map against genomes, you kind of propagate these bad models. So you have to be careful with the database you use. So sometimes you see this very like different protein length distribution. So then you have to check what happened, why it happened, and then maybe like have to go back and fix your annotation. And also like gene length, like model length and structure, or like the number of introns, size of these introns, exons, UTRs. Usually we compare to close related genomes and see if they kind of following the same pattern. If it's not, so why it happened? It can be, for example, you're annotating your genome with a very bad transcriptome, or it doesn't, or transcriptome is not coming from your strain, is like a different strain, and you see this, the mapping is not so good, so you're probably getting less UTRs than what you expect if you get like the transcriptome from your strain. So yeah, everything you have to check if it's like comparable to all the other close related genomes, and if it's not, why that, if you can explain that, then it's okay. But we have to be careful with all this metrics. And other ones like gene model support, like for example, it's supported by how many like PFAM domains. If it's supported by RNA-seq, like what is the percentage of the mapping? We blast against like SwissPro, NR, so we can see the if this data set is like it's supported by this. For example, for NR, since NR has like almost everything, usually we see like a high coverage when you blast against NR. Most of them are already present there, so we check all those metrics to see if it's making sense. And lastly, we check Sigma. Sigma is like kind of deprecated right now. People are not using that much, which is like this core gene that is found in all organisms. The Sigma, I think, is like 400 and something genes, but it was built like in the 90s, I guess, or 2000s. It's very old, so it's mostly from these model organisms, but they didn't like evolve. They didn't had anything new like in several years. And then another group created this program called Busco, which is very interesting. So now we implemented also a Busco here. So it was like just the beginning of this year. So we have another page for Busco where we blast, we compare this with several different data sets and to see this completeness. But Sigma, even being like old, it's very good. Usually when you see like it's missing some of the Sigma genes, when you do Busco, you see the same pattern. It's kind of faster and simple way to address this completeness. But working with Busco afterwards is always good. So this is another topic we look at, this completeness of this genomes. And now we're gonna go through the portals per se, just showing some of this information, how you can assess it and to understand, because we try to make it like simple to find information. But if you have any questions, you can always like contact us and send email, or even in the website you can log in and go to contact us, and then we always like respond all the questions, because we have these two portals. We have the Mycocosm and Phycocosm. So Mycocosm, we have all the fungi, and Phycocosm, we have all the algae. So here we're trying to show the data. We have some visualization methods, some analysis you can also do with this database, and also like download the information if you want to do yourself in your computer. So what we try to do with these portals is explore these gene models like in several different ways, and then also compare them, because the tricky part usually is to compare all these data sets. So the first step, of course, I strongly suggest you to sign in, create a password, put an email, create a password and log in, because it's good, because it's a good way to learn what genomes you are interested in, and what type of analysis, what type of data you're more interested. So it's a good way for us to track what's happening. And of course, once you will sign in, you can ask for help if you're not understanding something, or if you need some data, or if something's not working. You can always contact us, the button contact us, and then we always answer the questions and try to solve the problem as fast as possible. So what the genome portal, when you once you enter to Mycocosm, for example, and you're interested in some specific species, for example, you just click here and type the genome you're looking at, and then you're gonna show, like, for example, I'm showing here the Phanerochaeticus, but you can look for Aspergillus or whatever a genome, or for algae you can go like Chlamydomonas. So you're gonna show everything's available, or you can just click in these icons here. It's gonna also show all the genomes available for at each of these clades. And once you go there, you go to the genome portal. Genome portal is the front page of this genome. So usually it starts in the home page. So the home page has like a small text explaining everything about that fungi or the algae. So where it was isolated, why they're important, all this details, some reference, some genome reference, if it was published or not, and other types of reference. And usually the PI, they send like a picture to illustrate the portal. And so this tab here is very important. So everything you do is you can find here in this upper tab. So the first, as I told you, is the home page, which starts the descriptive image and text. And then starting from left to right, you see the search, where the search is very powerful. So you can look for example, you're already working with this genome for some collaborator like working with this genome, and I send you the data. You can look for the protein ID, which is unique for each genome. And then you're looking this protein ID or transcript ID, or you want to look for a function like, oh, I have this CAG pathway I want to look at, or this COG, or this gene ontology, or this specific Kzyme, or a PFAM domain. You can look for functional domains also, not only like transcript ID. And then the second tab is a blast. So it's pretty much like the NCBI blast. You click, but you're going to blast against this specific genome. So you can blast against the genes, like the same thing. You can do blast 10, blast P, blast X. So we have all these flavors, and you can look for specific gene set or proteins, or blast against the assembly, or only in the transcriptome, which is very cool because we provide also the Trinity assembly from the transcriptome. So sometimes it was happening in PacBio genomes where when you look for the gene within the genome, you were not finding it. And then when you blast against the transcriptome, you see the gene there. So it's probably something wrong with the assembly. It was, for some reason, it was kind of common in PacBio. I think it's not common anymore, but in the past, in the first iterations of PacBio, we were finding these cases. So that's why it's important to have the blast against also the transcriptome, not only the genome. Then you have the genome browser. I'll show it later, but it's a page where you can browse within the genome, see the genes, the positions, or where the tracks, where the genes are coming from, all this information. And after that, this is, I think, where people more like frequently go, is the annotations, where you can get the gene ontology, like PFAM domains, CAG, COG, the secondary metabolism clusters. Here, this one we use SMURF, a modified version of SMURF for secondary metabolism. We compared it with, how's it called, the other one, Anti-Smash, and we found this SMURF, this modified version of SMURF, is slightly better than Anti-Smash. So that's why we decided to use this method, and it's very clear. The results is very clear. You can see the cluster. So if you're interested in secondary metabolism, I strongly recommend you to go to some of these known genomes that produce secondary metabolites and take a look at these clusters. And then we have Kzymes, which is a better version because this is a manual annotation of Kzymes. We have a collaboration with Bernard here in South for France. So he does all these annotations, has a lot of people experts in Kzymes. So they curate this data set. Is not automatic annotation as other methods like DBCAN does. This one is a more curated. So I would say this one is, you can trust it better, the Kzymes we have at JGI, than other methods, because other methods pretty much only blast HMM and they're all automatic, and they just like get some thresholds and that's it. That's the only thing they do. Here we have a more curated prediction for these Kzymes. And we also have like peptidides, transporters, and transcription factors also. So this peptidase is only it's we get information from MEROPS, so that's what the database we use to predict these peptidases. And then transporters is from I forgot the name of the database for transporters. Yeah, I had it in my mind, but it slipped. If I remember, I'll talk it later. And then transcription factor is just a subset of PFAMs that are transcription factors. It's also a curated set of these genes that we know there are transcription factors. So there's no like specific database we go. It's more so like this well curated PFAM domains that are known to be transcription factors. And then we have this MCL clusters, which is pretty much the ortho MCL analysis, but we have a way to display, which is way more cooler than when you do it in your computer, which is very hard to parse. This case is very clear if you want to get only single copy genes or what is the present only in one genome. This kind of stuff is way easier to do here. I'll show it later. And then we have the Synteny tab, where you see the whole genome alignment with other genomes here. And then the download page. So if you want to download all this information, all the like assembly, GFF to work with your transcriptome data, or get the functional annotations. What else? Like all the information we have in this other tabs, you can get them on this download page. And then we have the info, where the info is more like if you want to look at the metrics of the genome, like the sequence assembly information, like how many scaffolds, N50, L50, like how many genes, all this, which collaborator like sequenced this genome. So you can go to all this page and see all this information. And then you have the status, which is pretty much showing the proposal and the project information. So if you send a proposal to JGI and then you get approved and you send us the sample and the sequence, here you're going to see like your information about your project that sequenced that genome. So if you want to learn like who did that or who was the responsible for that specific genome, you can just go to the status page and see the project. And help is like if you want to help to, they have a lot of tutorials of other pages like explaining how to use some of these tools. So you can go to this help page here. So here I'm showing an example when you go to the search page. For example, if you want to look for like this hydrophobin, which is very common, like protein in fungi. If you put hydrophobin and search, you're going to show all hydrophobins present in this genome. So here you can change the methods for search on the keywords. You can change the parameters of this the search. And here, when you go like across, it goes to default. The default is the default track, which is the filtered models, that the basic filter models where you have all the filtered genes. But you can change here and put like all, where you look all the predictions for that genome. So it's going to be like hundreds of thousands sometimes of gene predictions for this genome. But usually we left like default as the default here, because you want to look in this filtered models set. And then what type of search, the type of terms, this is more like for the, if you want like exact term or for similar, how to look at it. And what you get, the results is like all this in blue, there are links, so you can click and go to their specific page. Like if you click here, you go to the home page of that specific portal. The protein ID goes to a protein page, which I'm going to talk about later. The transcript ID, the same thing, is the page with the transcript information about this transcript. And the location, you're going to go to genome browser, where you're going to see like the location, like within the genome, where it's located, which genes are neighbors, all this kind of things. So, and you can see all of them. So if you're interested in one specific protein, or if you're interested, for example, in some geos or interproscan or PFAM, you can do the same thing. You just put it here and it's going to look it for you, because these links, you go to external databases. When you click in this geo, for example, it's going to go to the geo page for that specific case here, like the cell wall, for example. And we also have like manual annotations. So for example, if you're working with a specific gene in your lab, and you know that this gene has this specific function, or you found something different, or even the structure is a little bit different, you can do like this manual annotation. So here's an example of like this person like Ursula Cruz. She's like was studying this organism, and she found that this is a class 1 hydrophobin. So she went to this protein here specifically and added this information showing that this is this type of protein. So you also have this user annotation. You can implement your own annotation there. So the genome browser, just this example. So it's a very cool interface where you can like walk like different directions as any regular browser, or put the position specifically that you want to look at here, or like zoom in, zoom out, look at the bases or the type of amino acid, and if you want like fit, like just show that's a specific region. So we have a lot of this navigation tools. It's pretty much as the same as all other other genome browsers. And we have all these different lines here. We call them tracks. So you have the first one is like the GC content, and then you see like nucleotide conservation when you compare it to other fungi. So this is pre-calculated, so you can't change it yourself. For example, when we annotate the genome, we select what would be this comparators here. So because it takes a lot of time to calculate all this homology, that's why all of them are already pre-calculated. You can't do it yourself like on the fly. And then you see the conservation, how these genes are conserved, and see like based on the colors means like there's a gene present here in blue, for example. If it's white, because there's no like gene in this area here. And you have like introns and exons in different colors. So yeah, you can see how these genes are conserved when you do this comparison. And then you have the expression level. So this is based on the transcriptome, the person who performed the sequencing sent to us. Because what we do here is not expression level for one specific condition. So usually we ask the researcher, whoever like is doing this experiment, to grow the organism in several different conditions, because what you want here is like a diversity in transcriptions and to map it against the transcriptome and to predict, because this is pretty much a data that we use to support genome annotation. So it's not to see like differential expression. So usually it's a mix of different conditions. So what we do here, we see just like if it's highly expressed or not. What is a mix of conditions, not one specific. So you have the expression level of these genes. So it's good to see if the gene you're interested in, if they are expressed or not. So you can go here and see if there's some coverage and it's, oh, this gene is expressed. So it's probably true, because sometimes you see some genes that is kind of vague if it's true or not. So it's a good way to check if your gene is true or not. And this is the filtered models I was talking about. So it's the final set where it selects based on all the gene models. For example, you see a loci here, and if you look down, you see a lot of different gene predictors predicted different structures. So when you do the filtering, we check all of them. You have this heuristic to choose what is the best model for that loci, and then we show them in this the final filter set models. But we see that there's a lot of other gene models. Sometimes we don't select the correct one. It happens. It's not the rule. It's not so frequent, but it happens sometimes. And we can do this change. Like, for example, oh, this is not the gene model correct. This one is the correct one. So you can just demote this one and put the correct one there. So that's one thing that we can do. Also, as a user, you can just log in, and then you also can do this modification. Say, oh, this is the correct gene model. So it's something that the users also can do. And the last one here is the transcriptome coverage. Is similar to expression level, but is more, it's not quantitative, it's qualitative. It just shows that, this gene here, we have this coverage in transcriptome. Instead of here, you see like kind of this curve showing the gene expressed or not. And in the left side, you can reorder. For example, I don't like this gene set here. I like it like in the down, like in the bottom part of the page. So you can just click these arrows here and then put it down, or just hide. You can just click the minus and hide it, and its I, you can click and see more information about this track, like some details about this gene predictor, all this details. You click in this I, you get this information. So going like to the right, you have like when you click the annotations, I'm showing here the K-sign, but all these annotations, they have like a similar page. Only the only difference is the type of annotation you're looking at. So for K-zymes, it's very cool because you can look across different species. So all these numbers here are clickable. So if you click, for example, you want the auxiliary activities for the critical sporium, and you click here, you're going to see all these proteins described like one per line. So you can see the protein ID of this gene, for example, the location of the genome, the size of the gene, the protein size, what is the domain you find there, the CAS annotation, what the domain you can find there, the structure of this model. Like you can see, like these numbers here are the size. For example, this pink here is the exon, and then the intron. So you see the size of each exon and intron. So you can see the structure of this gene, and you click domains, you're going to see the domain, location of the domain within the protein. So then you can address, like, depending on your interest, you can see, oh, this one has two domains, not just one. You have like sometimes a carbohydrate domain, a carbohydrate binding domain, and a GH, or other combinations of different domains. So you can see in this page here. And then going to the other part, we have the MCL clusters. So it's pretty much what OrthoMCL or OrthoFinder or all these other tools do. But here we have this interface to show. So here we have the comparators. We have all these genomes in the comparison, and we have these donut plots where the colors shows like specific domains within the cluster, and then the thickness, the abundance relative to the largest one. So if you see like a full, the full one, because it's the largest cluster, with the largest species with more genes within the cluster, and then you're gonna getting thin because it's getting less genes. So you see a lot of these examples here, and then you have some domain, some cluster that have a lot of proteins with different domains. So you can see the proportion of each domain within the cluster. So this is a very cool method. And other thing is interesting is this small window here where you see any, for example. You can choose the number of genes you're going to see. For example, if you click any and put like 1, it's going to show all the clusters that have 1 in all the genomes. So then you, for example, you want to do like phylogeny, you want only the single copy genes, you can just put 1 here, and you're going to show 1 for all of them. Or if you want, oh, I want to see the genes that are present only in phenylketonucleotide crystal and not in others, you can do the same thing. You go here and put 0, so it's going to show 0 for everything. And in the phenylketonucleotide, you put like 1+, showing everything that have more than 1, and then you see the genes that is present only in this genome. So you can do a lot of different things with this window here. And when you click in the cluster, for example, click on the number 7, it's going to show a similar page as I showed before, but for the cluster. It'll show all the proteins present in this cluster, and 1 protein by line, the same thing. You can see the model as I showed before, the domains again for each of these proteins. And what we have extra here is the synteny, where you see the synteny of this specific gene here in this cluster, and what are the genes that are around this genome, this in this cluster. And when you look at, like, each color means it's ortholog. For example, you see this this pink here, they are the same gene, so they're just like two different in the different directions, but they're all neighboring this gene. You have a duplication here, so it's the same domain. So you can see, like, if the neighbor of this gene, the neighborhood of this gene is the synthetic or not. This is another very cool piece. So if you go to a different cluster, you're going to see this synteny page for that specific cluster. So you can look at the neighborhood of this on any cluster that you want. So it's a very cool page. So, and finally, I'm showing this protein page. So protein page, you always land here. For example, if you're, oh, I'm interested in this cluster, I have this gene here that I'm interested in. If you click on this number here, it's going to go to the protein page. If you're in the search and then you look for like a specific domain or specific function you're interested in, and then you see there's a protein number there. If you click there, it's going to land in this protein page. So here you have all the information about that specific protein. So you have the name. Usually the name shows the what gene predicted this gene. So this one is an EST extension using GeneWise prediction. And then we have the protein ID, so you'll be the unique identifier for this protein within the genome. The location, this one is clickable. You can click here and go to the browser page to see this gene in context. What is strand, a number of exons, all this, like what is the blast hit for this gene, and how many hits you found on NR, and some functional things like geo or interpro, and the description of this interpro. And here we have this view and modify manual annotation. This is the part where you can add your own annotation. So to do that, you have to be logged in. And then when you click, you go to a page where you can write down, oh, this is a protein X, Y, Z, or whatever. You can add your own annotation about this protein if you have it, for example, in your lab, you're studying this, and you can implement here. And you have all this other clicks here. You just see like the nucleotide 3' translation. So it's more like a browser see. Did you see like what is the translation of that specific region? You can perform your own NCBI blast, but here we already have a pre-computed blast results. So we have this too, the structure of the gene. So here the same thing, we see like UTRs in blue, exons in red, and then like a dash, you see the introns. So you see the structure, the size, and if you click here, you can see the sequence of this gene. So if you want to copy for whatever, like make a primer or whatever, you can just click here and then see the sequence and copy the sequence you want. And the domains present in this protein and the position of this domain. And here we have the protein sequence. So again, here you can click and then you can see the amino acid sequence of this protein. And we put like this small dash here to show that's the position of the intron where it collapsed. And the blast, usually that's interesting, because if you look at your protein, you want to see, oh, what is the function that is probably more related to? And then you see the blast results already here. So you don't need to go and blast yourself. You already see the NR blast in this page. Yeah, so I already talked about all these things here. And one other interesting thing here, you can go to the phylogeny tree. It's an interactive tree. So when you're in the home page or the Mycocosm page, you can go to any links there and click to see the tree for that specific clade. And this tree you can expand, collapse. You can click here and see like all the genomes of these groups. So you can do a lot of you can see a lot of information in the context of the phylogenetic tree. Like you can see the assembly line, gene counts, if it's published or not on the paper here. So you can see like the whole phylogenetic tree, like for all the fungi or all the algae, or you can just look for specific groups. So this is another very interesting feature to look at. And the new addition we have is the mitochondria. This one is not public yet. It's just showing you that we built like a mitochondrial annotation pipeline recently. So we have our own annotation. So and then we're planning to add this tag here, like mitochondrion, where you can see the mitochondria in all the genes, the annotation. So you can go to the download page and also download the mitochondrial annotation. I think soon, like in the next months, we're gonna see this this other tag here for fungal. But we're also planning to add this to algae, and algae not only mitochondria but plastids. But we're still kind of struggling to get this annotations from algae, because it's very complex genomes to assemble. So sometimes it's hard to get the plastid and the mitochondria. So we have only for some of them, but I think in the future we're planning to also add this information, which is very cool. So my already talking for like more than an hour, so just the take-home messages here. So I talk a little bit about the science we do at JGI. We're trying to explore this fungal diversity and specifically lignocellulose degradation. I showed some example of this white, brown, and gray rot fungi, some early diverging good fungi they're very good in degrading this plant biomass, the single cell and this the fungus that are not cultivable, and then 1,000 genome projects where we're trying to address this huge, like more diversity within the fungal kingdom. And also we are working toward the algal strain improvement. So we're trying to bring more genomes to the Mycocosm. So if you have, for example, algal genomes in your lab, your sequence, you have the annotation, you want to bring it to JGI, just contact us. We can do it easily. Actually, you want to bring more diversity to both Mycocosm and Mycocosm, because if you noticed, all this comparative analysis only makes sense when you have more diversity. If you have just a few of them, it's not so interesting. When you have like a big diversity, then you can find like signals, more genes that are present here and not there, and then you can find this more interesting things to explore. So I strongly encourage you to, if you have like your own projects in algo, you can just contact us. We can figure out a way to bring it to Mycocosm, or even help you to write a proposal to submit to Mycocosm. And also we for algal, we have this multi-omics pipeline. So we're trying to not only genome, but metabolomes, and try to mix them, to combine them, which is very difficult, and find the correct, like the metabolic pathways responsible for that specific metabolite. So we're trying to combine them and get a greater perspective of what's happening in this algal genomes. And also talk about this, the resources we have for comparative genomics, the Mycocosm and Mycocosm. So everything I talked about Mycocosm, it also works for Mycocosm. We just changed the genomes, but the idea is the same. You can look for example, the specific portals, or you can write down like the specific species, or you can go from phylogeny. For example, we have like, for fungi, I want to look for Polyporales or Aspergillus or other types of groups that you're interested in. You can just type the group here. You're going to go to that page. And for Mycocosm, the same. If you want to look for Chlorophyta, you can just put it there, or just go to the Mycocosm page, search, and then look for the specific genomes. And again, I strongly encourage you to register if you want to use our tools, because then you can help us to understand what people are interested in, and also you can get more help. And I think it's good to and learn the size of the community, because we know that sometimes it's because algal community it's kind of growing right now, but so you're trying to understand that and what people are needs to explore this types of data set. So this is my contact. If you guys are have any questions, or get in touch to understand more about the CSPs, or even how to bring data or how to explore more data at JGI, just you can contact me anytime. I'm happy to respond. And we have a Twitter account for Mycocosm. I don't think we have Mycocosm yet, but you can, if you have like, but we also put some information about the algae we have here on Mycocosm until we have the different account. Okay, thank you for the time. I'm open for questions.
Thank you very much, Andrei. It was a fascinating talk. I'm sure we have all learned a lot. And it's mind-blowing to see how much we had advanced through in silico analysis, and how broad it is, the applications for studying microbial systems. And we have many compliments and also several questions for you. So we'll display them here. So Raul is asking, since GC content is intimately related to DNA stability, what are the strategies these AT-rich fungi use to regulate their metabolism, prevent DNA degradation, etc.?
That's a good question. I don't study specifically these organisms. We have these collaborators. Michelle O'Malley, I think, is the one responsible for this organism, but I believe it's something related to the environment, because usually they are, these clumps, they like have a lot of bacteria around, so they kind of, and that's very hard to cultivate also. So I think it's, I'm not sure if they're, is the required oxygen. Yeah, because this is not my project, so, but these are good questions. I don't know exactly how they
are very interesting
prevent this.
microbes indeed.
Yeah.
Really curious. Okay, so to the next question. So Diogo, Dr. Diogo Tschoeke is asking how the neural network was trained to identify pathogenic and saprophagy fungi, only the presence and absence of these clusters.
Yeah, this one I know. Actually, because we use support vector machines, this work, we tried like several different approaches for, it's not a neural network, it's more like a simple, for this case, we use a simple SVM using all the clusters. So usually it's more like, this guys here is more like present absence. If you have, like this, we found that the presence of these genes, when you use only these specific clusters, if you use the combination of these 6 genes, you get like a very accurate prediction for genomes that you don't know if they're pathogen or not. So yeah.
Great, thank you very much. So the next question is, okay, so it's made by Taina. She's asking, in your opinion, what are the greatest challenges we face when translating omics data into the physiological context of the microorganism in their environments? How can we approach potential biases?
Yeah. Yeah, that's a good question, because yeah, I'm actually facing that. So I think the biggest bias is when you transfer things that are happening in the environment to a lab environment. So, and that part is very tricky to deal with. So I think the best way would be trying to simulate better the natural environment, and try not to simplify too much, because sometimes it's very common, and it's required. I understand sometimes you have to simplify to get some meaningful results. So yeah, that's
especially in transcriptomics. Do you think there is a good correlation that we can make nowadays to make sure that we are actually seeing the actual physiologic context, for example, of fungi or bacteria?
Mm-hmm. Yeah, actually the trickiest part, in my understanding, when you try to combine data from different sources, for example, you're working in one organism, then you have people working the same organism, but they have a different lab, a different environment, different thing, and then you try to combine this information. So because you have a lot of lab bias, like some things that people do, like some specific kits they use to extract that DNA or RNA or whatever. So, and then you have this bias, and when you put them together, things gets like messy. It's hard to gather information, is usually because the source of the data is different. So it's hard to address what was the problem to begin
So
with.
the main problem will be like the experimental
Yeah.
base problems.
Yeah.
Okay.
Yeah, I believe that when you work with bioinformatics, it's very common to see problems that happen like in the beginning, and then you try to fix using bioinformatics, which doesn't happen. If you have like a bad data in the beginning, like a bad experimental data, it doesn't matter. Your results will be bad after that. So making like a good like experimental analysis in the beginning, especially when you have the organisms, all these things, it helps a lot when you're trying to understand, especially this physiological context, or even combine like different types of data. Like I showed this multiomics, where you have like this lipidomics combined with transcriptomics. So usually it's very good if you have everything coming from the same lab, same conditions, and then you get you do this experiments in parallel, and then you extract the RNA for one and the lipidomics like in the same context here, and then you combine them. When you have like they're separate, like different labs, different things, and try to combine, and it's hard to make sense of this.
Perfect. So basically, just to summarize, it would be something like taking care about the experiments that you're going to proceed, and also combining this to different omics approaches, just to make sure that you're actually seeing what you're getting from the results.
Yeah.
Perfect. Okay, great. Thank you very much. So João is asking, so for eukaryotes, specifically in microbial consortia, we must use a combination of short and long reads?
You can do... what do you mean consortia? When you have like a mix of different eukaryotes and you want to separate them? Because
I believe yes, or different microbes perhaps, like bacteria, fungi, or fungi.
Yeah, because usually, because if you want to study them like all together, usually always like long reads are easier to separate things because you have like longer reads. For example, if you want to get like MAGs, this metagenomics assembled genomes, long reads is better because the method we use to separate them are coverage and Kamer profiles. So if you have these two metrics, like longer reads is usually better, because if you have only short reads, even if after you assemble it, if it's still like in short reads, it's impossible to know where they're coming from. So having the long reads usually is more helpful to get this separation of what genome are coming from, and then do like usually like bins based on coverage and then the Kamer profile, and then sometimes you can get even like complete genomes from this bins. And at least in my understanding, for this, the PacBio are more like long reads are more is easier to deal with.
But in your perspective, like from a bioinformatician perspective, is it better when you have like both short and long reads?
No, at least here, at least at JGI, I don't remember the last time I've seen data that is combination of long and short reads. Usually, because the good thing about PacBio, for example, if you have like more than the technology now is really way better than before. It was before the CCS technology. So if you get like more than 20x, usually you already have very good quality. Because back in the past, it wasn't true. You need like 50, like 60x to get like reasonable quality. But right now, you can get like lower coverage and get like good quality. So, and in the past, they were using short reads to cover and to remap and then fix the bases, like the SNPs. But I think right now we're not using it anymore. So I don't think it's that much advantage to do both, unless you have a purpose to do both sequencing, but I don't think it's necessary.
This is a very good advice. So are there any more questions? I believe there are. I don't know. Okay, so Taina is asking, does methylation profile in fungi correlates well with gene expression?
Only specific methylation and specific fungi. For example, that example I showed you before is for early divergent fungi, and only for that 6MA, the adenine methylation. And in that case, yes. But if you go outside of other like fungi, it doesn't happen. Like for Dicaria, which are like the most common fungi, they don't have this type of methylation. It has, but it's not so frequent, and they didn't see any difference in expression. But for early divergent, yes. So they're very different in this manner. So it's not like for all fungi. It's mostly for that specific clade and that specific type of methylation.
Perfect. Thank you very much. Okay, another one. Is it possible to check the interaction between pathogenic consortium and drugs in target?
Interaction between pathogenic consortium.
I believe that will be like the combination of more than one microbe.
Yeah. If it's, for example, if you want to look at you're gonna sequence them, like or genome or transcriptome, and then use like presence absence of the drug, for example. I think you can do that, especially if you're using like long reads. It's easier to address like the origin of this pathogen, like a fungi or whatever. Yes, because the tricky part here is like understand where, when you have like this consortium, where this read is coming from. So I think it's doable, but not trivial.
Yeah, sure. I agree. Okay, the next one. So I guess that was it with the questions, yes. So Andrei, thank you very much for being here with us today. That was a remarkable ending for our event, definitely. And we really hope you can join us in the future as well for the next events that the Anchor will hold. So thank you very much for the fascinating talk. It was amazing to hear you talking about the GGI work. It's an amazing work, so congratulations for it. And thank you very much again.
Yeah, thank you for the invitation. It was very nice. So if you have any questions, anything, just contact me. I have a LinkedIn profile, forgot to put it there, but if you put my name, there you're gonna see we can like talk, chat, and if you need any help with Mycocosm or Mycocosm, I can like direct you how to do an analysis or even like write a proposal, because usually if you talk to us before what you have, the data set you have, like the organism you have, it's easier to go like to the right path, how to write things and what we are interested in, because we are financed by DOE, so usually we have to follow their mission, what they're interested in. So it's important. We can help on that also. So yeah, and thank you for everything, and thank you for the questions. Yeah.
Yes, so guys, take this opportunity and write down his email and contacts so you can catch up with Andrei's work. So thank you very much again, Andrei.
Okay, thank you.
Okay, so we are on the end. We finally reached the end of our event. We would like to thank everybody for the presence and our amazing speakers. They definitely enlightened this couple of days of the Anchor event. I'm passing the word to Diogo for the final words. Thank you very much to your presence here, and I hope you have all enjoyed our event.
Okay, thank you, Juliana. So with that, we reach the end of this today Biotech Bioinformatic Tools and Applications workshop. I'd just like to remind everyone that with this event, we aim to be inclusive, embracing and engaging, and with that promoting the exchange of knowledge across the entire Atlantic in the field of marine biotechnology. If you want, you can watch back the recordings of the workshop right there on YouTube, and on our side we will also develop a written report about this workshop with a summary of the presentations and the main conclusions of the workshop. So look out for that as well in the near future. We have more activities planned, like I mentioned in the beginning of the session. The next Biotech Mar event will take place on June 27th and 28th, but you'll be able to find out more about it soon. In the meantime, you can check the Anchor website, which is allatlanticocean.org, where you can find more information about the project and also about the Biotech Mar Joint Pilot Action. So yeah, stay tuned because we'll have more news soon for you. Before we go, I'd just like to thank all of the speakers we had both today and yesterday. Thank you all for sharing with us all of your amazing work. It was really insightful. And thanks also to everyone watching on YouTube. I thank you as well. Thank you for your interactions in the chat. Thank you for your questions. I hope you had a great time both today and yesterday. Lastly, I'd like to thank the organization, Professor Krieger, Professor Fabiano, Kaique, Juliana, Jessie, Larissa. If I'm forgetting someone, I'm sorry. But thank you for your amazing work in creating this workshop. For me, that's all. Thank you, everyone. Have a great day, have a great weekend, and we hope to see you soon. Thank you.
Yeah. I would like to thank again all the speakers and the viewers and everybody that was involved on the preparation, organization. So I thank you a lot, and I'll see you guys next time.
I thank you very much.